From the surface, there is almost nothing to suggest what is below. The Western Pacific can look impossibly ordinary. Water stretches to every horizon. Swells move beneath the sky. Clouds pass overhead. Somewhere beyond sight lie Guam and the long curve of the Mariana Islands. But from a ship there is no visible edge, no scar in the sea, no warning that the ground beneath the water is about to fall away farther than almost anywhere else on Earth. If the ocean vanished for a moment, the landscape underneath would be difficult to recognize as part of the same planet. There would be mountains, volcanic ridges, broad plains, and immense slopes. And east of the Mariana Islands, the seafloor would descend into a long, crescent-shaped depression running for more than 2,500 kilometers through the Western Pacific. Near its southern end, within a region known as Challenger Deep, the bottom reaches roughly 10,935 meters below mean sea level, according to one of the most precise modern estimates. That is almost 11 kilometers of ocean above the seabed. It is tempting to stop there, because 11 kilometers already sounds like the whole story. It isn't. The Mariana Trench is often presented as though it were a single vertical hole in the ocean floor, with Challenger Deep sitting neatly at the bottom. That picture is useful for a diagram, but it creates the wrong mental image. The trench is an enormous geological landscape. Challenger Deep is one exceptionally deep region within it, not another name for the entire trench. Even Challenger Deep itself is not simply one smooth pit with one obvious lowest point. It contains irregular terrain and separate deep basins, and determining which tiny patch deserves the title of deepest becomes a surprisingly difficult scientific problem. That is why the number attached to Challenger Deep has changed. Over the years, expeditions have reported depths that differ by tens of meters. At first, this can sound strange. Humans can measure the height of mountains to remarkable precision. Satellites can track movements of Earth's surface from space. Why should it be so difficult to decide how deep the deepest part of our own ocean is? The answer begins with the fact that scientists are not looking directly at the ground. There are almost 11 kilometers of seawater in the way. A research ship moving across Challenger Deep cannot simply point a laser downward and read a number. Modern vessels commonly use multi-beam sonar, sending pulses of sound toward the seafloor and measuring how long the echoes take to return. It is extraordinarily powerful technology. A ship can sweep broad strips of terrain and build three-dimensional maps of valleys, ridges, and slopes that nobody on board can see. But sound does not travel through the ocean at one perfectly fixed speed. Its velocity changes with temperature, pressure, and salinity. The ship itself is moving. The sea surface is moving. The angle of the sonar beams matters. The exact position of the vessel matters. Even Earth's gravity has to be considered when extremely precise depth estimates are derived from pressure measurements. In a 2021 analysis based on submersible transects, researchers calculated a maximum observed Challenger Deep depth of 10,935 meters, with an uncertainty of about 6 meters at the 95% confidence level. Their corrections included the properties of the water column, atmospheric pressure, local gravity, and water level effects. 6 meters may sound like a large margin when talking about a point on a map. At almost 11 kilometers beneath a moving ocean, it is remarkably small. There is another problem. To find the deepest point, you first have to pass over it. Imagine trying to locate the lowest spot in an enormous mountain valley while flying high above it in darkness, using echoes to reconstruct the terrain underneath. If your survey lines are too widely spaced, a narrow depression may fall between them. If another expedition returns with better instruments and tighter coverage, the map can change. This is one reason the phrase, "the deepest point on Earth," is more complicated than it first appears. It sounds like a permanent pin that can be dropped onto a map. In reality, it is a measurement built from instruments, corrections, survey coverage, and definitions. And the Mariana Trench is only one example of a much larger problem. We have global maps of the seafloor. Satellites can infer broad underwater features because large mountains and trenches subtly affect gravity and even the shape of the sea surface above them. But those maps do not show the ocean floor with anything close to the detail people are accustomed to seeing on land. As of April 2026, only about 28.7% of the global seafloor had been mapped to modern high-resolution standards using technologies such as multi-beam sonar. That means more than 70% still lacks that level of detailed modern mapping. And even mapped should not be confused with explored. A sonar image can reveal the shape of a ridge without telling us what animals live on it. It can show a depression without revealing what is inside its sediment. It can identify a cliff without showing the microbial communities coating the rock. Mapping tells scientists where interesting places may be. Exploration requires something else. Cameras, landers, remotely operated vehicles, autonomous vehicles, sampling equipment, or in rare cases, humans. The deeper the seafloor becomes, the harder all of that gets. There is a practical reason for this that has nothing to do with mystery and everything to do with scale. A mapping vessel must move methodically across the ocean while its sonar sweeps the bottom beneath it. The deeper the water, the wider an individual sonar swath may become, but achieving useful resolution still takes ship time, fuel, specialist crews, and favorable conditions. NOAA has noted that if a single vessel attempted to map the entire ocean floor at all depths, the job would take on the order of 1,000 years. This helps explain one of the strangest things about modern geography. We live on a planet whose surface is mostly ocean, yet the most detailed physical landscapes familiar to us are almost all above water. We have walked across deserts, climbed mountains, drilled into ice sheets, and built roads through forests. A person can stand at the edge of the Grand Canyon and understand its scale with their own eyes. Nothing equivalent is possible at the Mariana Trench. There is no viewpoint, no cliff edge, no place where a visitor can stand and look into it. The entire structure is hidden beneath kilometers of water. Even its location can be misleading when imagined on a flat map. The Mariana Islands form an arc through the western Pacific, north of Guam and far from the large continental landmasses of Asia. The trench runs roughly parallel to that island chain on its eastern side. It is not a tiny isolated crack. It extends across a distance comparable to traveling between major European capitals several countries apart. Some sections are much shallower than Challenger Deep. Other depressions, including Serena Deep, descend to extraordinary depths of their own. The famous 10.9-kilometer figure belongs to the deepest known part, not to the entire trench floor. And even the word floor is misleading. There are slopes, scarps, sediment-filled depressions, faulted surfaces, regions disturbed by earthquakes, sections shaped by material sliding downslope. The bottom has topography of its own, hidden beneath a column of water so deep that Mount Everest could be placed inside Challenger Deep, and its summit would still remain more than 2 kilometers below the surface. NOAA lists Everest at 8,849 meters and Challenger Deep at about 10,935 meters. That comparison is repeated so often that it can lose its meaning. So picture it differently. If Everest stood where Challenger Deep is, nobody at the surface would see a mountain rising from the ocean. They could sail directly above the summit and still have more than 2 kilometers of water underneath the hull. And Everest would only represent the upper part of the journey. Everything below its summit would still be hidden inside the trench. This is the scale researchers work with when they send an instrument downward. A device does not simply go deep. It leaves almost every familiar ocean environment behind. It passes through kilometers of water before it even reaches the depth of an ordinary abyssal plain. Only later does it enter the hadal world associated with the deepest trenches. From the surface, none of this exists visually. The waves above Challenger Deep can be no more dramatic than waves elsewhere in the Pacific. A storm can pass over it without revealing its shape. Whales can move through the water, thousands of meters above the bottom. Ships can cross the region without anyone on deck being able to tell when they have passed over one of the deepest places on the planet. In 2015, NOAA researchers lowered a hydrophone into Challenger Deep to listen to this supposedly remote environment. From near the deepest part of the ocean, the instrument recorded earthquakes, whale calls, an overhead typhoon, and even the sound of ship traffic. The bottom of the world was not acoustically separated from everything above it. That is an important detail, because the Mariana Trench is often described as another world. In many ways it is, but it is still connected to the planet above. Material falls into it, sound reaches it, earthquakes move through it, human pollution eventually finds its way toward it. The rocks beneath it are connected to processes occurring deep inside Earth. Its organisms depend, directly or indirectly, on chemistry and energy moving through systems far larger than the trench itself. Understanding the Mariana, therefore, requires more than measuring a number and finding the lowest coordinate. The depth is the first thing people notice because it is easy to compare. The real story begins when we ask what created that depth. The seafloor beneath the Western Pacific is not simply sagging downward under its own weight. One enormous piece of Earth's outer shell is being forced beneath another. Rock that once formed the floor of an ancient ocean is bending, fracturing, and descending back into the planet. Water, trapped inside minerals, is being carried downward with it. Earthquakes trace its path into the interior. Volcanoes rise on the other side. And the trench is the scar left at the surface where that descent begins. The rock descending beneath the Mariana Trench began its journey long before the trench existed. Much of the Pacific seafloor approaching the Marianas is extraordinarily old oceanic crust, created far away at ancient spreading centers and carried slowly across the planet ever since. For more than 100 million years, parts of this plate have been cooling as they traveled. That cooling matters. Newly formed oceanic lithosphere is hot and relatively buoyant. As it ages, it loses heat, contracts, thickens, and becomes denser. By the time the Pacific plate reaches the Mariana system, it is no longer young seafloor floating high above Earth's interior. It is old, cold lithosphere, arriving at a boundary where there is nowhere left for it to continue horizontally. The movement is almost impossible to notice on a human timescale. Plates generally travel only centimeters each year. A fingernail can grow faster, but geology has something humans do not: millions of years. A few centimeters repeated year after year can move an ocean basin. Continents can separate. Entire seas can open and close. And along the eastern side of the Mariana Islands, the Pacific plate has reached a boundary with the Philippine Sea Plate, where those accumulated centimeters have produced one of the most dramatic landscapes on Earth. The two plates are converging, and the Pacific plate is going underneath. Geologists call this subduction. The word sounds almost too gentle for what is happening. One enormous slab of Earth's outer shell bends downward and begins descending hundreds of kilometers into the planet. The Mariana Trench marks the place where that descent begins. This also explains why the trench has its long, curved shape. It follows the boundary between the plates. Imagine taking a rigid board and slowly forcing one end beneath another. Before it disappears, the board has to bend. Oceanic lithosphere does something broadly similar, although on a scale of hundreds and thousands of kilometers, and with rock behaving very differently over geological time. As the Pacific plate approaches the Mariana boundary, its surface curves downward. The seafloor follows it, carrying the ocean above into an increasingly deep depression. The result is not a crack that opened and remained empty. It is a fold at the edge of a moving plate, continuously maintained because the plate itself continues to descend. The bending is violent at the scale of rock. The upper surface of the Pacific plate is stretched as it curves downward, producing faults that cut through the seafloor. Seamounts that spent millions of years traveling peacefully across the Pacific can arrive at the trench and be dragged into the subduction zone. Some are fractured, some deform the edge of the overriding plate as they pass underneath. Researchers studying the Mariana system have even documented material associated with ancient Pacific seamounts being caught up in this process. Reef limestone that once formed in a completely different environment can eventually become part of the geology of a deep subduction zone. That is one of the more extraordinary ways to think about the trench. The rocks entering it have histories. A piece of seafloor arriving at the Marianas today may have formed when the world above it looked nothing like the world we know. It may have traveled thousands of kilometers across the Pacific, accumulated sediment, carried extinct organisms, passed beneath countless storms and migrations, and finally reached a boundary where its journey across Earth's surface turns downward. But the Pacific Plate does not descend smoothly, like a conveyor belt disappearing into a machine. Rock catches, faults move, stress accumulates and is released. The descending plate can be traced into Earth's interior partly because earthquakes occur along it. Some are shallow, close to the plate boundary. Others originate far deeper, within the slab after it has already descended beneath the surface. Earthquakes therefore give geologists something almost like an X-ray of subduction. Plot their locations in 3 dimensions, and a dipping zone of seismic activity outlines the plate as it sinks beneath the Mariana region. This is why the trench cannot be understood as an isolated feature on the sea floor. The visible depression is only the upper edge of a structure extending far into Earth, and the process changes both plates. Between the trench and the chain of active Mariana volcanoes lies a region known as the forearc. It belongs to the overriding plate, positioned above the descending Pacific slab. At first glance, forearc sounds like another piece of geological vocabulary that only specialists need. But geographically it is simple. Start at the trench and move west toward the volcanic islands. The broad region you cross before reaching the active volcanic arc is the Mariana Forearc. NOAA maps it as the leading portion of the overriding Philippine Sea Plate, with the volcanic arc farther behind it. What happens underneath that region is far more interesting. The Pacific Plate is not carrying only dry rock into Earth. Oceanic crust has spent an immense amount of time interacting with seawater. Sediments lie on top of it. Water is stored in pores and incorporated into minerals. As the plate descends, temperature and pressure rise. Minerals that were stable closer to the surface begin to change. Water and other fluids can be released from the sinking slab and move upward into the material above. That movement of water is one of the hidden connections between a trench and a volcanic island chain. Farther from the trench, fluids released from the descending slab help alter the conditions in the mantle above it, promoting melting. Magma rises. Over geological time, repeated eruptions build volcanoes on the seafloor. Some grow high enough to become islands. This is why the Mariana Islands and the Mariana Trench sit in roughly parallel arcs. They are different expressions of the same subduction system. The trench marks where the Pacific Plate begins going down. The volcanic arc marks one of the consequences of what happens after it does. The distance between them represents an unseen journey beneath the crust. A drop of seawater cannot simply travel intact down the slab and then shoot into a volcano, of course. The real chemistry is much more complicated. Water becomes incorporated into minerals, released through reactions, moves through rock, and changes melting conditions. But the larger story is striking. Material that entered the oceanic plate at or near the seafloor can influence volcanic activity much later, after being transported deep beneath another plate. Even the forearc has its own strange response to these fluids. In parts of the Mariana system, water-rich fluids rising from the subduction zone react with mantle rocks in a process called serpentinization. The altered material can rise along faults and build enormous serpentinite mud volcanoes on the seafloor. Some contain pieces of rock and material that offer researchers rare clues about conditions much deeper in the subduction system. USGIS, work on cores from Mariana Forearc mud volcanoes, has identified material derived from the subducting Pacific Plate itself. In effect, geological processes can carry samples of a normally inaccessible plate boundary back towards scientists waiting at the seafloor. Later, these places will become important for another reason. The chemistry produced when water reacts with rock can create unusual environments for microbial life. But for now, what matters is that the trench, forearc, and volcanoes are connected parts of one machine. If we focused only on Challenger Deep, we would miss most of what the Mariana system is doing. There is still the question of why its southern end becomes so extraordinarily deep. Not every subduction zone creates an 11-kilometre trench. Even within the Mariana Trench, the depth changes dramatically from place to place. Challenger Deep is exceptional because several geological factors come together there. The incoming Pacific Plate is old, cold, and dense. The geometry of the plate boundary allows the slab to descend very steeply through much of the Mariana system. The overriding plate has also experienced extension rather than being pushed together in the simple way people sometimes imagine convergent boundaries. Behind the volcanic arc, parts of the crust have been pulled apart as the subduction system evolved. Near Challenger Deep, the structure becomes more complicated still as the Mariana boundary approaches the Yap Trench and the southern end of the arc curves sharply. The precise shape of Challenger Deep is therefore not produced by one simple cause. It is the accumulated result of plate age, bending, faulting, subduction geometry, and the tectonic history of the southern Mariana system. This is why calling it a hole misses so much. A hole suggests that something was removed. Challenger Deep exists because Earth's surface is being deformed while one plate is actively disappearing beneath another. And disappearing needs qualification. The Pacific Plate does not fall into some empty space below the crust. Earth has no enormous hollow chamber waiting beneath the trench. The slab enters the mantle, where increasing pressure and temperature progressively change its minerals and physical behavior. Some material becomes involved in processes that eventually contribute to magma generation. Other components may continue much deeper. Plate tectonics is, among other things, a planetary recycling system. New oceanic crust forms elsewhere at spreading ridges. Old oceanic lithosphere returns to Earth's interior at subduction zones. The Mariana Trench is one of the places where we can see the entrance to that recycling system. This recycling also gives the region a kind of restlessness that is easy to miss when looking at the calm ocean surface. The plates continue to move, faults continue to accommodate deformation, earthquakes continue, fluids continue to migrate through rock. Volcanoes of the Mariana Arc remain part of an active system. In the fore-arc, researchers study mud volcanoes and altered mantle material that have been brought upward from environments humans could never directly reach. The landscape is changing, although much of that change happens too slowly to watch and too far below the ocean to see. Occasionally, however, the system reminds us that geological time is not the only timescale involved. An earthquake can release accumulated strain in seconds. A submarine eruption can reshape part of the seafloor. A landslide can move sediment down slope. A seamount carried toward the trench can fracture the overriding region as it is forced beneath it. What looks from above like an empty patch of blue Pacific is sitting on top of a boundary where two enormous pieces of the planet are interacting continuously. Long before anyone measured Challenger Deep, all of this was already happening. The Pacific Plate was moving westward. The trench was evolving. Volcanoes were being built and destroyed. Islands appeared above the sea. Sediment collected in the depths. Earthquakes traced the descending slab into the planet. Humans lived beside this system without knowing its true dimensions because almost every important part of it was concealed by water. For most of history, there was no reliable way to see the landscape beneath a ship. Sailors could measure shallow water with a weighted line, especially near coasts where knowing the depth could mean the difference between safe passage and running aground. But the open Pacific was different. Lower a line into several kilometers of water and the problem becomes enormous. The rope has weight. Currents pull it sideways, a ship drifts. And even when the weight finally reaches the bottom, the people above must somehow know that it has arrived. For the Mariana Trench, that problem would eventually become one of the great measurement challenges of 19th century ocean science. A ship would arrive carrying no sonar, no satellite positioning, and no electronic map of the seafloor beneath it. Somewhere below was the deepest landscape humans would ever find. The only way to begin discovering it was to lower a weight over the side and let the line run. The only way to begin discovering it was to lower a weight over the side and let the line run. In the 19th century, that simple act was one of the few ways humans could investigate the deep ocean. There was no image of the seabed waiting on a screen, no satellite position showing exactly where the ship was, and no electronic instrument continuously measuring the distance below the hull. A sounding began with a heavy weight attached to a long line. The weight was dropped into the sea, and the crew paid out line as it sank. In shallow water, this was straightforward. In the open ocean, it became an exercise in patience and uncertainty. The line itself could weigh hundreds of kilograms. Currents pulled it sideways. A drifting ship could drag it away from the point directly underneath. The deeper the weight traveled, the harder it became to tell whether it was still falling or had finally touched the bottom. And once the measurement was made, kilometers of wet line had to be recovered. It was slow, physical work, performed over an ocean whose shape was almost entirely unknown. That was the world HMS Challenger entered when it left Britain in December 1872. Challenger had originally been a Royal Navy warship, but guns were removed and spaces were converted for scientific work. Laboratories and storage areas replaced some of the equipment of war. On board were naval officers, sailors, and a civilian scientific team led by Charles Wyville Thomson. Their voyage would last more than 3 years and cover roughly 127,000 kilometers, crossing the Atlantic, Southern, Indian, and Pacific Oceans. The ambition was enormous for its time. They were not simply trying to draw a deeper chart. They wanted to investigate the ocean as a system, its temperature, chemistry, currents, sediments, depths, and living organisms. The expedition would eventually collect thousands of specimens and help establish oceanography as a modern science. But every investigation depended on knowing how much water lay beneath the ship. Challenger carried a Bailey sounding machine and used Italian hemp line for its deep soundings. The line was about an inch around and, according to later historical descriptions of the equipment, weighed roughly 200 pounds for every nautical mile. At the lower end, detachable iron weights helped pull the apparatus downward. When the instrument reached the seabed, the weights could be released, making the long recovery easier, while a tube could retain a sample of the bottom. A depth measurement could therefore return something more valuable than a number. It could bring back a small physical piece of a world nobody aboard had seen. On March 23, 1875, Challenger was working in the Western Pacific near the Mariana Islands when the crew made one of those soundings. The line continued downward far beyond ordinary coastal depths. Eventually, the bottom was recorded at 4,475 fathoms, about 8,184 meters. The measurement was not Challenger deep as we define and map it today, and it was still more than 2.5 kilometers shallower than the deepest terrain later discovered. But in 1875, that distinction did not diminish what the crew had found. 8 kilometers of water was an astonishing result. The seafloor here was far deeper than the landscapes people knew on land could prepare them to imagine. More importantly, the sounding showed that the Western Pacific contained a depression whose true extent had not yet been measured. The expedition moved on. That was unavoidable. A 19th century research ship could not cover the seabed the way a modern mapping vessel can. Each deep sounding was essentially a single point. Between one point and the next could lie kilometers of completely unknown terrain. It was rather like trying to map an entire mountain range by lowering a tape measure from an aircraft at scattered locations, never seeing the ground between them. If the tape happened to land on a ridge, the landscape appeared shallower. If it happened to enter a valley, the number suddenly increased. A narrow depression could remain invisible simply because nobody measured in exactly the right place. Even so, Challenger transformed the way scientists thought about the ocean. The expedition demonstrated that the deep sea had structure, complex sediments, and abundant life. Its results eventually filled dozens of volumes, hundreds of new genera, and thousands of previously undescribed species were catalogued. The ocean was beginning to change from an empty blue area on maps into a physical environment that could be measured and studied. The technology used to measure it was already changing too. In 1872, the same year Challenger sailed, William Thomson, later Lord Kelvin, developed a practical wire-sounding machine. Thin metal wire created much less drag than bulky hemp rope, and allowed deeper measurements to be made more quickly and accurately. Challenger actually carried one of Thompson's machines, but the expedition continued using the familiar rope method for its soundings. In the decades that followed, wire increasingly replaced hemp. NOAA's historical accounts describe wireline systems as significantly faster and more accurate, and instruments using piano wire became important tools for mapping deep water. Yet even an excellent wire could only tell researchers about the point where it touched. The real revolution came when scientists realized that the bottom did not have to be physically touched at all. Sound could make the journey instead. Send a pulse downward. Wait. Detect the echo returning from the seabed. If the speed of sound through the water is known, the travel time reveals the distance. This sounds obvious now because sonar has become so closely associated with ocean exploration, but it fundamentally changed what a ship could do. Instead of stopping to lower kilometers of line and recover it, researchers could measure depth acoustically while moving. By the early 20th century, echo sounding was beginning to transform hydrography. Single-beam systems sent sound toward the seabed beneath a vessel and produced a line of depth measurements along the ship's route. The ocean floor was no longer being discovered one piece of rope at a time. It could begin to emerge as a continuous profile. In 1951, another British survey ship entered the Mariana region. Its name was HMS Challenger 2. More than 75 years had passed since the original expedition. The ship now had acoustic methods unavailable to its predecessor, and its survey found depths far beyond the 1875 measurement. Challenger 2 identified the extraordinarily deep depression that would become known as Challenger Deep, named for the ships that had helped reveal it. NOAA's historical record places its 1951 sounding at roughly 10.9 kilometers. That difference is worth considering. The original Challenger had found 8,184 meters. Challenger 2 found water more than 2 kilometers deeper in the same broad trench system. The trench had not suddenly deepened during those decades. Humans had simply become better at finding its deepest terrain, but sonar had its own limitations. A single beam still behaved somewhat like an invisible line dropped beneath the vessel. It measured what lay along the ship's track, leaving terrain between tracks less well known. The next major improvement was to stop asking the ocean one depth at a time. Multi-beam sonar sends out a fan of acoustic beams across a wide strip beneath the ship. As the vessel moves, those overlapping measurements build a swath of terrain. Pass after pass can eventually produce a detailed three-dimensional bathymetric map. In 1984, a Japanese survey vessel used multi-beam echo sounding in the Mariana Trench, part of the technological progression that allowed researchers to see the form of the bottom with a detail earlier explorers could not have imagined. Ridges became visible, slopes acquired shape, separate depressions could be distinguished. Challenger Deep began to look less like a number and more like a landscape, and with better measurements came an unexpected complication. The reported maximum depth kept changing. Different surveys produced figures such as 10,920 meters, 10,933 meters, 10,984 meters, and others nearby. Some historical measurements even exceeded 11,000 meters. It would be easy to look at those numbers and assume that somebody must have been wrong. The more interesting explanation is that measuring the deepest ocean pushes several technologies to their limits at once. An acoustic pulse traveling down through nearly 11 kilometers of water does not move through a perfectly uniform medium. Sound speed changes with temperature, salinity, and pressure. A beam sent at an angle travels differently from one aimed vertically. The ship moves and rolls at the surface. Its precise position must be known. The seabed itself is irregular. A survey that misses the lowest patch by a few hundred meters can return a different maximum from one that passes directly over it. Then there is the question of what depth is being measured relative to. Sea level itself moves with tides and atmospheric conditions. Gravity varies slightly from place to place. At the precision scientists now seek, effects that once would have been irrelevant begin to matter. Modern researchers can also measure depth using pressure sensors carried directly down into Challenger Deep. But converting an enormous pressure reading into an exact depth still requires corrections for the properties of the water and local gravity. In 2021, researchers published an analysis using data collected during submersible dives in 2020. Their deepest observed seafloor measurement was 10,935 meters below mean sea level, with an estimated uncertainty of plus or minus 6 meters at the 95% confidence level. The calculation accounted for water column properties, atmospheric pressure, gravity gradients, and water level effects. That uncertainty is not an embarrassment attached to the result. It is part of the result. Science becomes more reliable when researchers state how precisely something can actually be known, rather than presenting every measurement as perfect. There is also no reason to assume that one number permanently ends the search. Better instruments, denser survey coverage, and improved corrections can refine the picture again. What remained constant through all of these generations was the same basic problem faced by the sailors on Challenger. The ocean stood between the observer and the thing being measured. In 1875, the solution was a hemp line disappearing over the side of a ship. Later it was wire, then single acoustic pulses, then fans of sonar beams sweeping kilometers of terrain. Eventually, pressure sensors and acoustic altimeters traveled to the bottom themselves. Every technological step revealed more, but every improvement also showed how much the previous method had missed. And by the middle of the 20th century, the measurements had revealed something that created an entirely different problem. Challenger Deep was no longer simply a mysterious depression somewhere beneath the Pacific. Scientists now understood that its floor lay nearly 11 kilometers below the surface. They could estimate its shape, they could send sound to it and receive an echo. But an echo could not tell them what it felt like to descend through that water. It could not show what happened to a machine as pressure climbed through hundreds of atmospheres. It could not explain what kinds of animals could survive there, or whether there was life at the bottom at all. For the first time, humans knew approximately where the deepest known seafloor was. The next challenge was no longer to send a line down to it. It was to follow. And if we could somehow leave the ship above Challenger Deep and travel downward without a vehicle around us, the first part of the journey would feel surprisingly familiar. At the surface, sunlight penetrates the water. Waves mix oxygen into the upper ocean. Microscopic phytoplankton use sunlight to build organic matter, creating the foundation for food webs that eventually support animals thousands of times larger than themselves. This upper layer contains much of the ocean life humans recognize, and for roughly the first 200 meters, there is still enough sunlight for photosynthesis. Then the character of the ocean begins to change. Below about 200 meters lies what oceanographers often call the twilight zone. Daylight has not vanished completely, but it weakens rapidly. Reds and oranges have already been absorbed higher in the water column. Blue travels farther, giving the remaining light a cold, increasingly monochrome quality. By 1,000 meters, sunlight has effectively disappeared. From this point onward, there is no sunrise and no sunset. The only natural flashes may come from living organisms producing their own light. Yet, after descending an entire kilometer, we have completed less than 1/10 of the journey to Challenger Deep. The darkness arrives early. The pressure has been building from the moment we left the surface. At sea level, our bodies live under roughly 1 atmosphere of pressure from the air above us. We barely notice it because the pressure inside our bodies is balanced against it. Underwater, the weight of the water adds approximately another atmosphere for every 10 meters of descent. At 100 meters, the surrounding pressure is already around 11 times atmospheric pressure at the surface. At 1,000 meters, it is roughly 100 times greater. Continue to 6,000 meters, and NOAA gives the pressure at about 596 atmospheres. Pressure can be difficult to picture because it is invisible, and the usual language used to describe it can accidentally create the wrong image. The water is not simply pressing down from above like a giant weight sitting on a roof. In a fluid, pressure acts in every direction. An object at depth is being squeezed from above, below, and from every side. This is why the shape of a deep-sea vehicle matters so much. A flat wall or a sharp corner creates places where stress can become concentrated. A sphere distributes external pressure much more evenly, which is why the part of a deep submersible containing people is so often spherical. As we continue downward, the water also becomes colder. Sunlight does more than illuminate the upper ocean. It warms it. Below the surface layers, temperatures fall sharply through a region called the thermocline, before settling into the cold conditions typical of the deep sea. NOAA gives the average temperature of the deep ocean at about 4 degrees Celsius, though exact temperatures vary with location and depth. By 2,000 meters, we are traveling through a world that has no direct experience of daylight. By 4,000 meters, we are approaching the depths of broad abyssal plains found across much of the ocean. The names oceanographers give these layers can make them sound like tidy horizontal floors in a building, but nature does not draw perfectly sharp boundaries. They are useful descriptions of changing environmental conditions. The bathypelagic, often called the midnight zone, extends roughly from 1,000 to 4,000 meters. Beneath it, the abyssal zone reaches towards 6,000 meters. And then, at around 6,000 meters, something geographically unusual begins. Most of the ocean simply does not get any deeper. To continue downward, we need a trench. The environment below approximately 6,000 meters is known as the hadal zone, named after Hades, the underworld of Greek mythology. It is an evocative name, but what makes the hadal zone scientifically distinctive is not mythology. It is geography. Hadal environments occur mainly inside deep ocean trenches and a few other exceptionally deep depressions. They are separated from one another by much shallower seafloor, creating something like deep islands turned upside down. Instead of isolated mountaintops rising above a plain, these are isolated valleys descending beneath it. An animal adapted to life at 8,000 meters in one trench cannot simply travel horizontally to another trench while remaining at the same depth. Between them may lie thousands of kilometers of shallower ocean. That isolation will become important when we encounter the animals living here. But first, the machinery carrying us downward has to survive. At 6,000 meters, the pressure is already severe enough to crush ordinary equipment. A camera taken from the surface cannot simply be waterproofed and lowered into the trench. If it contains an air-filled cavity, the surrounding water is trying to collapse that cavity with hundreds of atmospheres of pressure. The same is true of batteries, computers, connectors, motors, and sensors. Deep-sea engineers therefore have two broad choices. They can resist the pressure, or they can allow it in. Components that must remain at ordinary internal pressure can be placed inside thick housings made from materials such as titanium, glass, or ceramics. Other systems can be designed to tolerate ambient pressure, sometimes by filling them with incompressible oil, so there is no large pressure difference between inside and outside. James Cameron's Deep-Sea Challenger, for example, used pressure-compensated lithium-ion battery systems immersed in silicone oil, rather than enclosing every battery inside an enormous heavy-pressure vessel. Every solution creates another problem. A stronger pressure housing usually means more material. More material means more mass. More mass requires more buoyancy to stop the vehicle from sinking uncontrollably. More buoyancy material adds size. Size increases drag and can increase the energy needed to maneuver. Engineering for the deepest ocean, therefore, becomes a careful negotiation between strength, weight, buoyancy, power, and reliability. At ordinary depths, a small design inefficiency may be inconvenient. At full ocean depth, it can determine whether the vehicle comes home. One of the strangest engineering problems is that the material keeping a vehicle afloat must itself survive the pressure. A surface ship floats because its hull encloses large volumes of air. Try that at 10,000 meters, and a conventional hollow structure would be catastrophically vulnerable. Deep vehicles instead use specialized buoyant materials designed to withstand compression. Woods Hole's robotic vehicle Nereus used around 1,500 hollow ceramic spheres for flotation, each only about 9 centimeters across. Ceramics were also used in some of its pressure housings, because conventional titanium solutions would have made the vehicle too heavy for its full ocean depth mission. That gives some sense of how unusual this environment becomes for engineers. Ceramic, a material most people associate with brittle household objects, can become part of a machine designed to descend to the deepest ocean because its compressive properties can be extremely useful when carefully manufactured into the right shape. Glass is similarly valuable. Thick glass spheres are routinely used to protect deep ocean electronics because a well-made sphere distributes pressure efficiently. But the tolerance for flaws becomes unforgiving. A pressure vessel is only as trustworthy as its material, its geometry, its welds, its openings, and the components passing through it. A tiny defect can become significant when the entire structure is loaded from every direction. This is why human-occupied spheres undergo extraordinary testing. When Woods Hole built a new titanium personnel sphere for the submersible Alvin, the finished sphere weighed around 11,000 pounds and was designed for operations to 6,500 meters. Before people were allowed to trust their lives to it, engineers placed it in a hydrostatic chamber and subjected it to pressures corresponding to 8,000 meters, while hundreds of measurements monitored how the metal responded. Challenger Deep lies roughly another 3 kilometers below even that test depth. Near its floor, pressure is around 1,000 times atmospheric pressure at the surface, exceeding 1,000 bar. The exact value varies with depth and water properties, but the scale is what matters. Every square centimeter of an exposed structure is experiencing an enormous compressive load. And unlike darkness or cold, pressure cannot be solved by adding a brighter lamp or a thicker layer of insulation. It determines the architecture of almost everything we send down. It is also why an implosion at great depth is so destructive. If an air-filled housing fails, the water outside does not slowly leak into a relaxed, empty space. There is a huge pressure difference between the ocean and the interior. Water accelerates inward as that difference collapses, and the structure can fail extremely rapidly. This is not a theoretical danger. In 2014, Nereus was lost while exploring the Mariana Trench northeast of New Zealand. The vehicle had reached about 10,000 meters when communication was lost. Debris later recovered at the surface indicated a catastrophic implosion. Nereus had already successfully reached Challenger Deep years earlier. It was an advanced vehicle designed specifically for hadal exploration. Its loss demonstrated something important. Reaching these depths once does not make them safe. The environment does not become less extreme because engineers have learned how to enter it. As we pass 8,000 meters, then 9,000, then 10,000, there is another distinction worth making. A machine containing air has to resist the ocean as an external force. A living animal is not built like a submarine. Most soft tissues contain large amounts of water, and water is far less compressible than gas. An animal without large gas-filled spaces does not experience pressure as though an invisible hand were simply crushing it flat. This is why deep-sea animals do not need titanium skeletons. But that does not mean pressure is harmless to them. Its effects move inward to a different scale. Pressure can alter proteins, membranes, and the chemical reactions on which cells depend. It can change how molecules behave. For life, the problem is not merely keeping the body from collapsing. It is keeping the machinery of the cell functioning when the physical environment is pushing that machinery away from the conditions under which most organisms evolved. This difference between machines and organisms is one of the reasons the deepest ocean becomes so interesting. Engineers survive pressure by resisting it with carefully designed materials, by equalizing it where possible, and by accepting enormous constraints on weight and shape. Evolution had millions of years to find entirely different solutions. By the time our imaginary descent approaches the bottom of Challenger Deep, sunlight has been absent for nearly 10 kilometers. The water is cold. The pressure is more than 1,000 times what we experience at the surface. A poorly designed hollow structure can be destroyed in an instant. Even some of the most sophisticated vehicles ever built have been lost trying to work at comparable depths. Yet when cameras and baited instruments finally arrived in the hadal trenches, they did not find an empty landscape. Something moved toward them. Something moved toward them. It was not the enormous predator that popular images of the deepest ocean might lead us to expect. Often it was something much smaller, pale, shrimp-like animals appearing out of the darkness, gathering around bait that had only recently arrived on the seafloor. These were amphipods, crustaceans related distantly to familiar beach hoppers. And in the hadal trenches, they can occur at depths where no fish has ever been confirmed alive. Their arrival revealed something important about the world we had just descended into. The bottom of a trench may be dark, cold, and subjected to enormous pressure, but it is not necessarily empty, and one of the most effective ways of discovering that is surprisingly simple. Scientists send down food. A hadal lander is built to make the journey without remaining attached to the ship by 11 kilometers of cable. It carries cameras, lights, instruments, and sometimes traps, then falls freely through the water under the weight of ballast. Hours later, it reaches the seabed. A piece of fish attached in front of the camera acts as bait. Then the lander waits. When the experiment is finished, an acoustic command from the ship tells it to release its ballast. Buoyant material carries the equipment back toward the surface, with its recordings and samples. The method has transformed hadal biology because the animals are observed where they actually live, rather than being dragged through kilometers of changing pressure inside a net. And when these cameras began spending more time in trenches, the old picture of the deepest ocean as a nearly sterile place became increasingly difficult to defend. Noah notes that before 2014, much of what scientists knew about hadal life still rested on major sampling campaigns conducted in the 1950s by the Danish Galathea and Soviet Vityaz expeditions. Those voyages had already demonstrated that life existed at extreme depth, but the sampling was too scattered to answer many of the ecological questions researchers now ask. How populations change with depth, whether animals move between trenches, why one species dominates one location and disappears from another. Modern landers began filling in some of those gaps, and what they recorded was not simply a collection of organisms enduring terrible conditions. It was an ecosystem organized around an unusual problem. Almost nothing grows here. At the surface, sunlight provides the energy for phytoplankton to photosynthesize. That newly produced organic matter enters the food web. 11 kilometers lower, there is no photosynthesis. No meadow of deep-sea plants waits at Challenger Deep. Much of the food supporting animals there ultimately began somewhere far overhead, often as microscopic life in sunlit water. When organisms near the surface die, when zooplankton produce waste, when fragments of tissue, mucus, shells, and other organic particles begin sinking, they join a slow downward rain known as marine snow. The name makes it sound delicate. Ecologically, it is a supply line. Some particles take weeks to reach the deep sea floor. Much of their useful energy is consumed or decomposed during the descent, so only a fraction of what begins near the surface survives the journey into the deep ocean. This makes the geography of a trench unexpectedly important. Picture an ordinary abyssal plain as a vast, relatively open floor several kilometers beneath the ocean. Now cut a steep, V-shaped depression through it. Organic particles falling directly from above can enter the trench, but material can also move downslope. Sediments shift. Earthquakes and slope failures can redistribute organic matter. The walls and shape of the trench influence where material eventually accumulates. Rather than behaving only as an extreme food desert, some trench axes can act as traps for organic carbon. That does not make Challenger Deep abundant in the way a coral reef is abundant. It means the deepest landscape has ways of concentrating the limited resources arriving there, and animals have evolved to exploit them. A large carcass changes the situation dramatically. If a fish, squid, or marine mammal dies higher in the ocean and enough of its body reaches the deep seabed, it represents an enormous pulse of energy compared with the steady trickle of marine snow. Scavengers that might normally search over large areas can respond to the chemical traces of food. This is why baited cameras can seem to transform an apparently empty patch of sediment. At first, the light may reveal almost nothing. Then amphipods arrive. More follow. Soon dozens or hundreds can crowd around the bait, feeding with an urgency that makes sense in a world where the next large meal may be unpredictable. Some hadal amphipods have become especially interesting because their bodies demonstrate just how thoroughly life can adapt to depth. Species differ between trenches and depth bands, and many hadal animals appear to have restricted distributions. The trenches are separated from one another by shallower seafloor, so each can behave partly like an ecological island. An animal specialized for 8,000 meters cannot necessarily cross thousands of kilometers at 4,000 meters to colonize another trench. Isolation gives evolution room to take different paths. Amphipods are only part of what the cameras find. Sea cucumbers move across deep sediment, feeding in a way that looks almost absurdly modest until we understand what they are doing. Many swallow seafloor mud and process the organic material attached to it, extracting nutrition from particles that have survived the long journey downward. They are effectively eating the sediment itself for the traces of food mixed through it. Their soft bodies also illustrate why the deepest animals often look so different from familiar surface species. They do not contain the large gas-filled spaces that would become disastrous under enormous pressure. Some are translucent, some appear almost gelatinous. Others have broad feeding structures or unusual body shapes suited to moving over soft sediment. Polycate worms occupy another part of this community. Some burrow, some scavenge or prey on smaller organisms. Their presence matters less because they look spectacular on camera, and more because a trench floor is not merely a surface with animals sitting on top of it. The sediment is habitat. Organisms live within it, process it and alter it. Beneath them, microorganisms carry out an even larger share of the chemical work, something we will eventually follow deeper into the mud. Then there are organisms that almost seem designed to confuse our expectations of what a single cell can be. In 2011, a team using free-falling landers in the Mariana Trench's Serena Deep recorded xenophyrophores at depths reaching 10,641 meters. A xenophyrophore can resemble a sponge or a delicate piece of coral sitting on the seabed, but it is a giant single-celled protist. Individual cells can exceed 10 centimeters in size. A cell large enough to be clearly visible without a microscope sounds like an exception to one of biology's basic rules, yet these organisms can be abundant on parts of the deep seafloor. Their structures also create habitat for other organisms, making one enormous cell part of a wider community. Finding them so close to the deepest reaches of the Mariana system forced another adjustment in how scientists imagined extreme life. Pressure had not reduced everything to tiny, simple forms hidden invisibly in the sediment. A single cell could become larger than a human hand and function under pressures approaching those at Challenger Deep. This is where the phrase "survival under extreme pressure" can become misleading if we are not careful. These organisms are not spending every moment struggling against an environment that is trying to kill them. For a hadal amphipod born thousands of meters below the surface, one atmosphere would be the unusual condition. Its proteins, cell membranes, metabolism and body chemistry have been shaped by evolution in a high-pressure world. Bring many hadal organisms rapidly toward the surface, and the change can itself become destructive. What is extreme to us is normal to them. The more useful question is how evolution makes that normality possible. Part of the answer lies in body structure. Part lies in chemistry. Proteins must retain the right shapes to perform their functions. Cell membranes must remain usable rather than becoming too rigid or unstable. Enzymes still have to work. DNA must be copied and repaired. Every ordinary process of life continues while pressure alters the physical behavior of biological molecules. Different hadal organisms solve those problems in different ways, and some solutions appear to have limits. That becomes especially visible when we look at fish. Fish do live in trenches. In the Mariana Trench, one of the most important is Pseudoliparis suirae, the Mariana snailfish. It does not resemble the imaginary monster people might expect from one of Earth's most inaccessible environments. It is relatively small, pale and soft-bodied, with adaptations that allow it to live at extraordinary depth. Hadal snailfishes have been found in multiple geographically separated trenches, and studies of their ear stones, or otoliths, suggest individuals of studied hadal species may live for years rather than existing as short-lived curiosities at the edge of survival. Their success is one of the best examples of vertebrate life adapting to the hadal environment. But there is something peculiar about where they occur. Cameras descending through the upper hadal zone can find fish, continue deeper, and fish become rarer, go farther still, and they vanish from the recordings altogether, even while amphipods, sea cucumbers, and microorganisms continue below. NOAA summarizes current evidence for a physiological boundary somewhere around 8,200 to 8,400 meters, beneath which no fish has yet been confirmed living. Challenger Deep continues for roughly another 2.5 kilometers beyond that proposed boundary. So somewhere during our descent, long before we reach the deepest floor, an entire vertebrate body plan appears to run into a problem it cannot solve simply by evolving stronger bones or becoming softer. To understand why, we have to leave behind the scale of animals and look at what pressure is doing inside their cells. To understand why, we have to leave behind the scale of animals and look at what pressure is doing inside their cells. The Mariana snailfish is a useful place to begin because almost nothing about it suggests an animal living close to the known depth limit for vertebrate life. It has no armor, no enormous teeth, and no heavily reinforced skeleton. Its body is pale and partly translucent. Beneath the skin is a thick layer of gelatinous tissue containing more than 95% water. Its bones are thin and incompletely hardened. Its skull is not completely closed, and it has no swim bladder. Even its movement is economical. Recent observations of Mariana snailfish swimming in their natural environment show them gliding slowly with gentle movements of broad pectoral fins, their low-density bodies requiring relatively little effort to remain near the seabed. It looks fragile because, by the standards of a fish brought to the surface, it is. But judging it at the surface is almost like judging a polar animal in a desert. This body belongs to another physical world. Researchers have collected Pseudoleparis swirri from depths around 7 kilometers in the Mariana Trench, and cameras have observed the species across a wider hadal range. In 2019, scientists comparing its genome and anatomy with shallower relatives found changes associated with several of the problems created by extreme depth. Its reduced skeleton is one example. In ordinary vertebrates, mineralized bone provides strength and protection, but building a heavy, rigid skeleton is not necessarily advantageous for an animal that spends its entire life suspended in cold water under hundreds of atmospheres of pressure. The Mariana snailfish has unusually thin and incompletely ossified bones, and researchers identified a mutation affecting a gene involved in bone mineralization that may help explain this reduced hardening. Its skull is also incompletely closed, allowing pressure to equalize rather than enclosing a rigid internal space. None of this means that bone itself is simply crushed by deep water. The adaptation is more subtle than that. A lightly mineralized skeleton reduces density, changes the mechanical properties of the body, and fits into a broader design in which there are few compressible spaces for pressure to act upon. The absence of a swim bladder is especially important. Many fishes closer to the surface use a gas-filled swim bladder to control buoyancy. Gas behaves badly when pressure changes. The deeper an air-filled space descends, the more it is compressed. A hadal snailfish avoids that problem entirely. Its buoyancy comes instead from watery, gelatinous tissues and a lightly built body. But the hardest problems begin at a scale we cannot see. Every cell is surrounded by a membrane, an extraordinarily thin barrier made largely from lipids and proteins. It controls what enters and leaves the cell, helps maintain chemical gradients, and contains machinery essential for life. Under increasing pressure, those membranes tend to become more tightly packed and less fluid. A membrane that becomes too rigid cannot function normally. Transport proteins embedded within it may stop working properly. The cell begins losing control over processes that are basic to being alive. Hadal fishes appear to compensate partly by changing the composition of their membranes. More unsaturated fatty acids can help membranes remain fluid under cold, high-pressure conditions. In the Mariana snailfish, researchers have found expansions in gene families associated with fatty acid metabolism and membrane transport. The animal is not resisting the ocean with some biological equivalent of a pressure vessel. Its cells have been altered so that the machinery inside them continues working while exposed to the same pressure as the surrounding water. Proteins create another problem. A protein is not simply a chain of molecules. It must fold into a particular three-dimensional shape before it can perform its job. Enzymes depend on their shapes to control chemical reactions. Structural proteins need the correct form to support tissues. Proteins that transport substances through membranes must open, close, and bind to other molecules in precise ways. High hydrostatic pressure can interfere with these structures and reactions. Evolution therefore needs a way to keep proteins stable without making them so rigid that they stop functioning. One of the most important pieces of that solution is a small molecule with a complicated name, trimethylamine N-oxide, usually shortened to TMAO. TMAO occurs naturally in marine animals and helps stabilize proteins against forces that would otherwise disrupt their structure. Researchers studying fishes from different depths noticed a remarkable pattern. The deeper the fish normally lived, the more TMAO tended to accumulate in its tissues. The relationship was sufficiently consistent that it offered a biochemical explanation for how fishes tolerate increasing pressure. As depth increases, pressure destabilizes proteins more strongly. More TMAO helps counteract that effect. Mariana snailfish possess genetic changes associated with the production of TMAO, fitting the same general pattern. At first, this sounds like an elegant solution with no obvious ending. If pressure increases, simply produce more TMAO. Go deeper, produce more again. Given enough evolutionary time, perhaps a fish could eventually descend all the way to Challenger Deep, but chemistry imposes a cost. A fish cannot fill its cells with unlimited amounts of TMAO. The molecule also contributes to the concentration of dissolved substances inside body fluids. At sufficiently high concentrations, maintaining the necessary balance between the inside of a fish and the surrounding seawater becomes increasingly difficult. Based on measurements across deep-sea fishes, researchers have proposed that this trade-off creates a physiological depth limit somewhere around 8,200 to 8,400 meters. It is not a wall in the water. There is no sudden line where a fish crossing a few meters farther down would instantly die. It is better understood as a region where the biochemical solutions used by fishes become increasingly difficult to sustain, and the observations are remarkably consistent with the prediction. The deepest confirmed fish sighting currently comes from the Izu Ogasawara Trench south of Japan, where a snailfish was filmed at 8,336 meters. Other fish have been recorded at similarly extraordinary depths, and older trawl records contain some uncertainty because an animal caught by a net cannot always be assigned to the deepest point reached by that net. But despite decades of increasingly capable cameras and landers descending into the deepest trenches, no fish has been reliably documented living at 9,000 meters. None has appeared at 10,000. None has been seen swimming across the floor of Challenger Deep. This is important because the absence cannot easily be blamed on scientists never looking there. Baited landers have repeatedly attracted scavengers at depths beyond the known fish limit. Amphipods arrive. Other invertebrates appear. The cameras work. The bait is detectable, but the fish do not come. Challenger Deep reaches roughly 10,935 meters, leaving about 2.5 kilometers of ocean between the proposed fish boundary and the deepest seabed. That lower section is not a small unexplored margin. It is an entire vertical environment roughly equivalent to placing several of Europe's tallest mountains one above another. Fish appear to be excluded from all of it. The idea becomes even more interesting when we remember what evolution has already accomplished. Snailfishes are an enormously successful family. Different species occur from shallow coastal environments to hadal trenches. Genetic work suggests the Mariana snailfish lineage diverged from a relatively shallow water relative on the order of 20 million years ago. Over that time, natural selection changed its pigmentation, skeleton, membranes, transport systems, and protein chemistry. Vision became less important in permanent darkness. Pigmentation was reduced. Its body became suited to neutral buoyancy without a gas bladder. Cellular systems changed in ways that help them function under extraordinary pressure. Yet, after all of those modifications, another few thousand meters may remain inaccessible because the obstacle is no longer something that can be solved by changing body shape. It lies in the chemistry shared by vertebrate cells. This does not mean scientists have proven that a fish can never be discovered below 8,400 meters. Biology is full of organisms that survived where previous assumptions said they should not. The deepest confirmed observation is a record, not an inviolable law. Sampling in hadal trenches is still sparse, and the exact position of any physiological boundary remains uncertain. A future expedition could extend the known range. What makes the proposed limit compelling is that two independent lines of evidence point toward roughly the same place. Biochemical predictions based on how fish regulate their cells under pressure, and actual observations showing fish disappearing as cameras descend through the deepest hadal environments. Below that depth, evolution continues working. It simply works with different bodies. Amphipods can live deeper. Sea cucumbers occur at extraordinary depths. Xenophyophores have been recorded beyond 10 kilometers in the Mariana system. None of them is a vertebrate trying to maintain exactly the same internal chemistry as a fish. Different branches of life have inherited different biological machinery, and that gives them different boundaries. The deepest ocean, therefore, does something unusual. It begins sorting life according to what its chemistry can tolerate. At 7 kilometers, a snailfish can be an active predator. Around 8 kilometers, fish are approaching the far edge of their known world. Continue toward 9, 10, and nearly 11 kilometers, and vertebrates disappear entirely. Yet, the sediment beneath the camera is anything but biologically empty. A single spoonful can contain organisms in numbers far greater than the handful of animals visible above it. They leave no silhouettes swimming through the lights. Most would never appear in even the clearest footage from a submersible. To find them, scientists had to stop looking across the seabed and begin looking into it. To find them, scientists had to stop looking across the seabed and begin looking into it. At Challenger Deep, that means taking something that appears almost disappointingly ordinary and treating it as one of the most valuable samples on the expedition: mud. A core recovered from nearly 11 kilometers beneath the surface may look like little more than dark sediment packed into a tube. There are no enormous jaws waiting inside it, no animal large enough to become the photograph from the expedition, and much of what matters cannot be seen at all. Yet a few centimeters of this sediment contain an ecosystem whose inhabitants outnumber the animals moving above it by an extraordinary margin. Bacteria and archaea occupy the spaces between sediment grains, living in a world measured not in kilometers, but in fractions of millimeters. They consume organic molecules, transform nitrogen compounds, process hydrocarbons, and interact with metals that have accumulated in the trench. Some live near the sediment surface, where oxygen carried by the bottom water can still penetrate. Others inhabit deeper layers, where oxygen disappears and entirely different forms of metabolism become useful. And because the floor of Challenger Deep acts partly as a collection basin, the microbial community at the bottom is not simply a deeper version of the community living on the slopes above it. The shape of the trench helps create different worlds inside the same trench. This became particularly clear when researchers compared sediments collected from the slopes of Challenger Deep with samples from its bottom axis. They reconstructed hundreds of microbial genomes from environmental DNA and found distinct communities with different metabolic abilities. Of the microbial genetic signatures examined in one major study, around 26% were sufficiently unfamiliar to be considered novel at the level being analyzed, and the amount of novelty generally increased with water depth and depth within the sediment. In other words, The farther researchers moved into this environment, the less familiar its microscopic inhabitants became. That does not mean 1/4 of the organisms were completely new forms of life, unlike anything biology had encountered before. Genetic novelty is more precise than that. Many belong to broader groups scientists already knew, but their genetic sequences were sufficiently different to suggest poorly characterized or previously unknown lineages. It is the microbial equivalent of entering a forest, where you recognize that something is a tree, but cannot identify many of the species. The difference is that here, the forest is invisible. Scientists reconstruct it from DNA scattered through the mud. This is where modern genetics changed deep ocean exploration almost as dramatically as sonar once changed mapping. A traditional expedition could collect an organism, bring it into a laboratory, and try to grow it. That remains enormously useful, but most environmental microorganisms do not conveniently grow under standard laboratory conditions. Some depend on chemical relationships with other species. Some reproduce slowly. Some require pressure, temperature, or nutrient conditions that are difficult to recreate. Metagenomics avoids part of that problem. Researchers extract DNA directly from an environmental sample and sequence the genetic material belonging to the entire community. Computational methods can then sort millions of fragments and reconstruct sections of genomes, sometimes producing remarkably complete genetic blueprints for organisms nobody has ever cultured or watched alive. The genes do more than reveal who may be there. They offer clues about what those organisms are capable of doing. In Challenger deep sediments, many of those capabilities revolve around one of the oldest problems in biology: finding something to eat. The trench floor receives organic material from above, but by the time that material has traveled through kilometers of water, much of its easiest energy has already been consumed. What arrives can include degraded remains of plankton, pieces of larger organisms, fecal material, and organic compounds attached to sinking particles. Sediment can also move down the steep trench slopes and accumulate along the axis. This creates a funnel effect. Challenger Deep is not simply deeper than the surrounding abyssal seafloor. Its topography can concentrate material transported from a much larger area. When researchers measured microbial activity in Challenger Deep sediments and compared it with a nearby site around 6,000 meters deep, they found something that initially seemed backwards. Biological oxygen consumption at the deepest site was roughly twice as high, and the sediments contained more microbial cells. Usually, biological activity on the seafloor declines as the ocean becomes deeper because less fresh organic material survives the journey from the surface. Challenger Deep broke that simple expectation. The trench appeared to be collecting enough organic matter to support intensified microbial activity even under pressures approaching 1,100 times those at the surface. This gives the Mariana Trench a role in a much larger planetary story. When microscopic organisms break down organic material in marine sediments, they help determine whether carbon is recycled back into the ocean or buried for longer periods. The amount contributed by one narrow trench is small compared with the entire ocean, but trenches are not chemically irrelevant holes. Their steep geometry, sediment transport, and unusual rates of deposition can create concentrated zones of carbon processing at extreme depth. And some of the molecules being processed there are unexpected. Researchers sampling water deeper than about 10,400 meters in Challenger Deep found an unusually large proportion of bacteria with the ability to degrade hydrocarbons. These microbes included groups known to consume aliphatic hydrocarbons, molecules built largely from chains of carbon and hydrogen. The obvious assumption might be that this must be evidence of petroleum pollution reaching the bottom of the ocean, but the story is more interesting. Hydrocarbons can be produced naturally by living organisms, and analyses of compounds in Mariana sediments suggested that at least some of those being consumed could have a biological origin. Scientists isolated bacteria from 10,400 meters and showed that some could break down alkanes under laboratory conditions designed to simulate aspects of the deep sea. More recent work has pushed the story beneath the seabed itself. A sediment core taken from roughly 10,816 meters and extending about 7.5 meters below the seafloor, revealed diverse genetic pathways associated with both aerobic and anaerobic hydrocarbon degradation. The deeper sediment contained microorganisms from numerous groups capable, at least genetically, of participating in a complicated hydrocarbon cycle. Some genes suggested degradation, others suggested the potential for hydrocarbon production. What initially looked like a strange concentration of oil-eating bacteria now appears to be part of a much more complex system of carbon compounds being produced, transformed, and consumed in the deepest sediments. Then there is arsenic. The word immediately suggests poison, and at sufficient concentrations it is. But microbes have been interacting with arsenic compounds for billions of years, long before humans gave the element its reputation. Challenger deep sediments can accumulate arsenic, delivered with sinking particles, and researchers have found concentrations in some hadal sediments substantially above those at non-hadal comparison sites. The microorganisms living there do not simply endure its presence. Genetic and RNA evidence indicates that some can transform arsenic compounds, and certain reactions may even be linked to gaining energy. This is a useful reminder of how different microbial life can be from the animals we know. An animal searches for food, consumes it, and extracts chemical energy from organic molecules. Microorganisms have access to a much wider collection of metabolic strategies. Depending on the species and environment, they can use compounds involving nitrogen, sulfur, iron, arsenic, and other elements in reactions that help power their cells. Recent research across deep-sea sediments, including the Mariana Trench, has found previously unrecognized genetic diversity associated with arsenic transformation, and evidence that some arsenite-oxidizing microorganisms may also contribute to carbon fixation. A substance that is toxic in one biological context can become part of an energy pathway in another. Nitrogen is being transformed here as well. Some microorganisms convert ammonia through nitrification, others perform denitrification, ultimately returning nitrogen to the atmosphere in gaseous form. Researchers have also detected genetic pathways for anaerobic ammonium oxidation, or anamox, particularly in parts of the bottom axis sediment. These processes matter because nitrogen is essential to proteins and DNA. The global nitrogen cycle is not controlled only by forests, farms, and sunlit ocean water. Some of it is being quietly processed by organisms buried in mud, almost 11 kilometers beneath the Pacific. The remarkable thing is that none of this announces itself visually. A camera can spend hours above Challenger Deep and record amphipods crossing the frame, while most of the biological activity beneath it remains completely hidden. The sediment may look motionless while oxygen is being consumed, organic carbon is being dismantled molecule by molecule, nitrogen compounds are being transformed, and arsenic is changing chemical form. Even the boundary between one microbial community and another can occur over a distance smaller than a human hand. Near the surface of a core, oxygen may support one set of reactions. Deeper down, as oxygen is exhausted, microbes capable of using other chemical pathways become more important. Each layer preserves a different balance of food, chemistry, and pressure history. This is why collecting deep sediment correctly is so difficult. The sample has traveled from an environment under more than 1,000 atmospheres of pressure into 1 atmosphere at the surface. Temperature changes, pressure changes, chemistry can change during recovery. Some organisms may become inactive or die. Researchers therefore use specialized corers, landers, in situ instruments, and pressure-retaining systems when possible, while also comparing DNA, RNA, chemical measurements, and direct measurements made on the seabed. DNA can show that a metabolic pathway may exist. RNA can help indicate which genes were being actively expressed. Chemical profiles show what substances are present or disappearing through the sediment. Oxygen sensors can measure respiration without requiring scientists to bring the entire environment into a laboratory. Each method reveals a different part of something no single camera could ever show. And this changes what it means to say that the Mariana Trench remains unexplored. Discovering another large animal would be spectacular, but some of the largest unknowns at Challenger Deep may never produce a dramatic silhouette in a submersible's lights. They may be genetic. A microorganism could contain an enzyme that functions under pressure in a way scientists have never seen. An unfamiliar archaeal lineage could be participating in a chemical cycle we have only partly reconstructed. Entire networks of organisms may be exchanging compounds inside sediments that have been sampled only a handful of times. In one genomic study of Challenger Deep sediments, researchers reconstructed 178 microbial genomes spanning 26 major groups, with 16 of those groups being reported from hadal sediment for the first time in that work. Another, broader investigation reconstructed 586 genomes from sediments across the slopes and bottom axis. Those numbers are not a census of everything living there. They are pieces recovered from a biological archive that remains vastly undersampled. A core only centimeters wide represents an almost absurdly small portion of Challenger Deep. Move that core hundreds of meters, and the chemistry may change. Move from the trench axis onto a slope, and the microbial community can change with it. Dig farther beneath the seabed, and another set of organisms appears. The deepest known point in the ocean is therefore not simply a surface waiting for explorers to photograph it. There is another frontier underneath that surface, extending downward into sediment and rock. By the middle of the 20th century, scientists could lower instruments into this world. They could retrieve mud, measure its chemistry, and eventually use genetics to expose life invisible to the eye. But there was one experiment no sediment core could reproduce. A machine could descend without fear. It could be lost and replaced. Sending human beings into the same pressure was something else entirely. And in January 1960, two men climbed into a steel sphere barely large enough to hold them, and prepared to find out whether a human could travel all the way to the bottom. And in January 1960, two men climbed into a steel sphere barely large enough to hold them and prepared to find out whether a human could travel all the way to the bottom. The morning of January 23 was not especially inviting. Challenger Deep lay hundreds of kilometers southwest of Guam, and the sea around the support ships was rough enough to damage parts of Trieste while it was being prepared. There were reasons to postpone. There were always reasons to postpone a dive like this. Instead, Lieutenant Don Walsh of the United States Navy and Swiss engineer Jacques Piccard entered the bathyscaphe and committed themselves to a journey no human being had completed before. Trieste looked nothing like the streamlined submarines people usually imagine. More than 15 meters long, it resembled a large floating tank with a small steel sphere attached underneath. That unusual shape came from an equally unusual idea developed by Jacques' father, Auguste Piccard. Auguste had already explored the opposite direction. In the early 1930s, he had risen into the stratosphere inside a pressurized gondola carried by a balloon. To go into the deepest ocean, he effectively reversed the concept. A bathyscaphe would be a kind of underwater balloon. Instead of a light gas lifting it through the atmosphere, a liquid less dense than seawater would provide buoyancy in the ocean. Trieste carried around 22,000 gallons of gasoline in its enormous float. Gasoline sounds like a strange choice for a deep-diving vehicle, but it solved an important problem. It is lighter than seawater, providing lift. Yet unlike air, it does not collapse dramatically as pressure increases. The men themselves were protected inside something very different. Beneath the float hung a steel pressure sphere just over 2 meters in diameter. Its walls were about 5 inches thick. The sphere had been manufactured by Krupp in Germany and installed after the United States Navy purchased Trieste in 1958, specifically because the earlier sphere was not designed for the full depth of Challenger Deep. The new one was rated for roughly 36,000 feet. There was little wasted room inside. Walsh and Piccard would spend the journey sitting almost shoulder to shoulder among instruments, valves, gauges, and communication equipment. They could not simply open a hatch if something went wrong. Once the dive began, kilometers of ocean would soon make that impossible. Trieste controlled its vertical movement with an elegant system that depended partly on gravity and electromagnets. Iron shot served as ballast. Holding that heavy iron made the bathyscaphe sink. Releasing it made the gasoline float sufficiently buoyant to pull the vehicle upward again. The ballast was held in place electromagnetically, which gave the design an important safety feature. If electrical power failed, the magnets would release the iron and Trieste should begin rising automatically. The vehicle did not need engines powerful enough to climb 11 kilometers through the water. It only needed to become lighter than the water around it. After the dive began, the Pacific slowly closed over the structure. The bright surface disappeared first, then the remaining daylight faded. The support ships above could communicate only acoustically, sending sound through the water. Radio waves that work so well through air do not travel usefully through kilometers of seawater. Every message therefore crossed an environment in which even communication behaved differently. The descent was slow enough that Walsh and Picard had time to experience the ocean changing around them rather than simply arriving at the bottom. Trieste sank at roughly a meter per second for much of the journey. The temperature outside fell. The gasoline contracted as it cooled, affecting buoyancy and requiring adjustments. At several depths, layers of water with different temperatures changed the vehicle's behavior. Inside the sphere, there was no panoramic view of the world outside. The men looked through small observation ports. Their lights illuminated only a limited volume of water surrounded by darkness. Most of the descent was therefore measured not by scenery, but by instruments. Depth increasing, pressure increasing, time passing. At 10,000 feet, they were already deeper than most submarines could ever travel. At 20,000 feet, they had entered the hadal environment. At 30,000 feet, they were approaching depths at which no human beings had ever existed. Outside the sphere, pressure was climbing toward 1,000 atmospheres. Inside, Walsh and Picard remained at approximately normal atmospheric pressure, separated from the ocean by those inches of steel. This distinction made Trieste fundamentally different from the soft-bodied animals we encountered earlier. A snailfish allows pressure to exist throughout its water-rich tissues and adapts its chemistry to function under it. Walsh and Picard could not. Their lungs contained gas. Their bodies depended on familiar surface pressure. The sphere, therefore, had to maintain almost the entire pressure difference between one atmosphere inside and roughly 1,000 outside. Somewhere around 30,000 feet, after hours of descending, there was a violent sound. Walsh later remembered it as a sharp crack that shook the cabin. At that depth, a sound like that is difficult to dismiss. There is no rescue vehicle waiting nearby. There is no emergency ascent that takes a few minutes. If the pressure sphere itself were failing, the men would have almost no time to understand what was happening. They checked their instruments. The sphere appeared intact. Nothing indicated that the ocean was entering the cabin. Trieste continued downward. The source of the noise would only become clear after the vehicle returned to the surface. A plexiglass window in the flooded entrance tube above the pressure sphere had cracked. Later Navy accounts concluded that the damage resulted from temperature effects rather than the pressure sphere failing. The entrance tube was intentionally flooded during the dive, so the cracked outer window did not expose Walsh and Picard directly to the surrounding pressure. They did not know all of that when they heard it. What they knew was that the vehicle still appeared to be functioning, and the bottom was now closer than the surface. They continued. After almost 5 hours of descent, the rate of sinking began to change. Sediment was approaching beneath them. At 1:06 in the afternoon, Trieste settled onto the floor of Challenger Deep. The depth indicator aboard the bathyscaphe showed 37,800 feet, although later corrections placed the actual depth closer to 35,800 feet, roughly 10.9 kilometers. The precise number would be revised again as later surveys improved the map, but the achievement did not depend on a few meters. 2 human beings were sitting at the deepest known seafloor on Earth. The landing itself immediately demonstrated how difficult it was to observe anything there. Trieste disturbed the fine sediment as it settled. A cloud of pale material rose around the sphere and reduced visibility. Picard described the bottom as flat and covered in light, soft ooze. They had travelled nearly 11 kilometres to see the seafloor and then partially obscured it simply by arriving. Their stay lasted only about 20 minutes. It was enough time to make observations, communicate their success to the surface, and look through the small window into a landscape that had never before contained human beings. And during those minutes came one observation that would survive for decades in accounts of the dive. Picard reported seeing what he believed was a flatfish, something resembling a sole, perhaps 30 centimeters long, lying on the seabed before slowly swimming away. At the time, the sighting seemed extraordinary. A bony fish living at the deepest point in the ocean would mean vertebrate life had somehow adapted to the full pressure of Challenger Deep. Picard regarded it as evidence that complex life existed even at the greatest ocean depths. Today, the observation is much harder to accept. As we have already seen, no fish has been reliably documented anywhere close to 11 kilometers. Modern physiological evidence suggests that fishes may reach a pressure-related limit somewhere around 8,200 to 8,400 meters, and the deepest confirmed fish observation is a snailfish at 8,336 meters. That leaves more than 2.5 kilometers between the deepest verified fish and the floor where Picard believed he saw one. There is also a basic anatomical problem. True flatfishes are not known from anything approaching Challenger Deep. Some researchers have suggested that what Picard saw may have been a sea cucumber or another pale benthic animal whose shape was difficult to interpret through a small window under artificial light, while sediment remained suspended around the vehicle. There is no photograph clear enough to settle the question. The moment remains part of the history of the dive, but not reliable evidence that a fish lives at Challenger Deep. In a way, that uncertainty makes the story more valuable rather than less. Trieste had achieved something extraordinary, yet even with human eyes physically present at the bottom, observation remained difficult. Reaching a place did not automatically mean understanding what was there. After roughly 20 minutes, Walsh and Picard prepared to leave. They released iron ballast. Freed from some of its weight, Trieste began the long ascent. Challenger Deep slowly disappeared beneath them. There was no engine roaring upward, no rapid escape from the pressure. The bathyscaphe simply became buoyant and rose through the water column, taking more than 3 hours to return to the surface. When daylight finally returned and Trieste emerged beside the support vessels, Walsh and Picard had been submerged for around 9 hours. Only then could the damage from the cracking sound be examined properly. The dive had proved that a human-occupied vehicle could reach the deepest known ocean floor and return. It had also demonstrated how narrow the margin could feel between success and failure. The pressure sphere, the gasoline float, the ballast, the seals, the windows and the communication systems had all needed to work through a journey that pushed 1950s engineering to an extraordinary extreme. President Dwight Eisenhower later presented Walsh and Picard with honors, and Trieste became one of the most famous vehicles in oceanographic history. But the most surprising part of the story is what did not happen afterward. There was no immediate succession of expeditions carrying scientists back to Challenger Deep. Trieste itself continued working for the Navy and later helped locate debris from the lost nuclear submarine USS Thresher. But humans did not return to the deepest seafloor the following year, or the decade after that, or even within the next half-century. For 52 years, the only people who had ever seen Challenger Deep with their own eyes remained the two men who had entered that cramped steel sphere in 1960. If the engineering problem had already been solved, at least once, the question was why doing it again proved so difficult. If the engineering problem had already been solved at least once, the question was why doing it again proved so difficult. Trieste had shown that Challenger Deep was reachable, but it had not shown that going there could become routine. The distinction mattered. A single successful descent could justify extraordinary preparation, a specialized vehicle, and enormous risk. Scientific exploration needed something different. Instruments that could return repeatedly, move across the seabed, collect samples from precise locations, and give researchers enough time to compare one part of the trench with another. Spending hours descending for 20 minutes on the bottom was an achievement. It was not yet an efficient way to study an ecosystem thousands of kilometers long. There was also the simple problem of money. Almost every component designed for full ocean depth had to function under pressures that excluded ordinary engineering solutions. A vehicle needed a support ship, a trained crew, launch and recovery systems, navigation, communications, pressure-resistant electronics, and enough redundancy that one failed component would not necessarily end the mission. A human-occupied vehicle added another requirement. Every failure had to be considered in terms of the people trapped inside it. Scientists increasingly realized that many questions could be approached without putting anyone there at all. For decades after Trieste, the next visitors to Challenger Deep were therefore machines. One of the most important came from Japan. In March 1995, JAMSTEC's remotely operated vehicle Kaiko descended to 10,911 meters in the Mariana Trench. Unlike Trieste, Kaiko did not carry a person. Its pilots remained aboard a ship at the surface while the vehicle worked below, connected through a sophisticated system that allowed commands and information to move between the two worlds. The result was a different kind of presence. Human eyes could inspect the bottom without human bodies having to survive its pressure. Kaiko returned in 1996 and collected sediment containing microorganisms from 10,898 meters, the first such samples recovered by an ROV from beyond 10,000 meters. In 1998, it collected the hadal amphipod Hirondelia gigas from Challenger Deep. Those missions represented a quiet change in what exploration meant. Piccard and Walsh had needed to be physically present to look through a window. Now a camera could become the window, and a robotic arm could become the hand. Yet removing people did not remove the danger to the machine. In 2003, Kaiko was working off Japan when its secondary cable broke and the vehicle was lost. The ocean had claimed one of the few machines on Earth capable of reaching its greatest depths. Japan continued developing hadal technology, including later Kaiko systems and the 11,000-meter class autonomous vehicle Abismo, because there was no simple replacement waiting on a shelf. Full ocean depth vehicles remained rare precisely because building one was so difficult. The United States encountered the same problem from another direction. Woods Hole Oceanographic Institution began developing a vehicle called Nereus in the early 2000s. Nereus was unusual because it could work in two different ways. As an autonomous vehicle, it could operate without a cable, following programmed instructions. For more complicated work, it could function as a remotely operated vehicle connected to the ship by a remarkably thin fiber optic tether. A conventional heavy cable, 11 kilometers long, creates enormous engineering problems of its own. It has weight, drag, and vulnerability to movement in the water. Nereus's lightweight system helped separate the vehicle from those limitations. Engineers also used unusual materials, including ceramic flotation spheres, to survive full ocean pressure without making the machine impossibly heavy. In May 2009, Nereus reached 10,902 meters in Challenger Deep. For the first time since Kaiko, scientists again had a robotic vehicle capable of working at the deepest seafloor. It could spend hours there while researchers remained safely above. It could collect samples, capture images, and operate instruments. This looked much closer to the future of hadal science than the heroic one-off descent of 1960. But Nereus also demonstrated how temporary that capability could be. 5 years later, during an expedition to the Kermadec Trench northeast of New Zealand, communication with the vehicle disappeared at 9,990 meters. The team waited for emergency systems to bring it back. Nothing surfaced. Debris was eventually found, indicating that part of Nereus had catastrophically imploded under pressure, approaching 16,000 pounds per square inch. An $8 million research vehicle had been destroyed in an environment it was specifically designed to explore. Nobody was injured because nobody had been inside it, and that was one of the great advantages of robotic exploration. Scientists could accept risks that would be unthinkable with a crew, but the loss also showed why the deepest trenches remained so difficult to study. Technology had advanced enormously since Trieste, yet the basic physical environment had not become any more forgiving. Then, in 2012, a human returned. James Cameron had spent years developing Deep-Sea Challenger with an engineering team in Australia. The vehicle looked almost nothing like Trieste. Instead of a broad gasoline float with a sphere hanging beneath it, Deep-Sea Challenger stood vertically in the water like a narrow green tower. Much of its structure consisted of specialized syntactic foam, a strong, buoyant material containing microscopic hollow glass spheres embedded in resin. The pilot occupied a small steel sphere near the bottom. There was barely enough room to move. Cameron would spend the dive with his knees bent, surrounded by controls and displays, looking through a small viewport. The shape was deliberate. Trieste had taken almost 5 hours to descend. Deep-Sea Challenger was designed to move vertically through the water much faster, so that more of the expedition could be spent actually exploring. On March 26, 2012, after rough weather and a series of increasingly deep test dives, Cameron entered the sphere and began descending alone. Challenger Deep arrived after about 2 hours and 36 minutes. 52 years after Walsh and Piccard, the third human being in history reached the deepest known seafloor, and the first did so alone. The difference in technology was enormous. Cameron carried high-definition and 3D cameras, powerful LED lighting, a robotic manipulator, a sediment sampler and instruments measuring pressure, temperature and salinity. Uncrewed landers were also operating as part of the wider expedition. For the first time, a human at Challenger Deep could do more than look through a window and wait to rise again. Yet the dive immediately reminded everyone how easily a carefully planned scientific program could shrink once the vehicle reached the bottom. Several thrusters failed. A hydraulic leak affected the manipulator system. Cameron had hoped to collect more biological material, but the equipment problems limited what he could retrieve. What he did see was striking for its emptiness. He described a soft, almost gelatinous plain and traveled across it toward a slope. There were no giant predators and no mysterious fish moving through his lights. The largest active animals he noticed were small amphipods. After the extraordinary engineering required to put a person there, the landscape itself appeared quiet, sparse, and almost lunar. But that apparent emptiness was partly an illusion, as our journey through the sediment has already shown. Much of Challenger Deep's biology was too small to see through a viewport. Cameron's expedition returned valuable images, measurements, and samples, and perhaps more importantly, it demonstrated that a modern, full ocean depth human vehicle could spend meaningful time moving across the seabed. Still, another one-off dive would not solve the larger problem. Scientists needed repetition. A place this large cannot be understood from a handful of visits, any more than the Amazon could be understood by landing a helicopter in one clearing. That next change came with a vehicle built around the idea that full ocean depth should be repeatable. The submersible limiting factor, developed for the 5 Deeps expedition and piloted extensively by explorer Victor Vescovo, used a titanium pressure hull designed for repeated dives to the deepest places in every ocean. In 2019, Vescovo descended into Challenger Deep as part of an expedition that combined human dives with landers and detailed sonar mapping. Crucially, the vehicle did not make one ceremonial visit and retire. It went down again. Different passengers could see the hadal environment. Scientists could participate. Observations could be repeated rather than treated as an event that might not happen again for decades. Full ocean depth diving was beginning, slowly, to shift from a record attempt into an operational capability. China was developing that capability as well. In November 2020, the crewed submersible Fendouge descended beyond 10,900 meters in Challenger Deep with 3 people aboard. Its program followed years of Chinese deep submergence development and added another vehicle capable of taking researchers into the hadal zone. Japan, meanwhile, had continued sending robotic systems, landers, and sampling equipment into deep trenches after the loss of Kaiko. No single country or vehicle had made the environment easy to reach, but the number of ways scientists could investigate it was finally increasing. And the most useful machines were not always the spectacular ones carrying people. free-falling landers could be deployed in groups. One could carry baited cameras, another could measure oxygen, another could collect water or sediment. They could sit on the bottom for hours or longer, then release ballast and return to the surface. Instead of placing one expensive vehicle at one location, researchers could begin building networks of observations across different depths. Modern multi-beam sonar could map the terrain above them. Pressure sensors could refine depth measurements. Genetic sequencing could extract biological information from the samples they recovered. Human submersibles, robotic vehicles, and relatively simple landers were beginning to complement one another rather than compete. This changed the kinds of questions scientists could ask. The first expeditions wanted to know whether anything could reach Challenger Deep. Later expeditions could ask why amphipod communities changed with depth, whether sediment chemistry differed between the trench axis and its slopes, how much organic carbon accumulated at the bottom, how microbial populations varied over distances of only a few kilometers, or whether separate depressions inside Challenger Deep were biologically connected. Repeat visits also exposed a problem hidden by the phrase, "We have explored the Mariana Trench." A submersible traveling a few kilometers across the bottom has explored a path only meters wide through a geological structure more than 2,500 kilometers long. A lander sees whatever happens within the small pool of light around its camera. A sediment core samples a patch of mud narrower than a dinner plate. Even dozens of successful dives amount to tiny windows into an enormous landscape. And as the maps became better, that landscape became harder to describe, with the familiar image of one V-shaped trench descending neatly to a single deepest point. Challenger Deep itself contained separate depressions, slopes, ridges, and sediment-covered terrain. Serena Deep lay elsewhere in the trench. Faults cut across the seabed. Landslides had moved material. The floor that early explorers could only imagine from a depth measurement was finally becoming visible as topography, and it looked nothing like a simple hole. The floor that early explorers could only imagine from a depth measurement was finally becoming visible as topography, and it looked nothing like a simple hole. If all the water above Challenger Deep could somehow disappear, there would be no perfectly circular shaft dropping vertically into the planet. Instead, we would find ourselves looking across a long, narrow valley surrounded by enormous slopes, broken ridges, scarps, faults, and broad areas where sediment has collected on the lowest ground. Even the name Challenger Deep refers to more than one depression. Modern high-resolution surveys reveal three main deep basins arranged along the trench axis, usually described as the western, central, and eastern pools. They lie kilometers apart. Each has its own shape and its own deepest terrain. This immediately creates a problem with one of the most familiar phrases in geography, the deepest point on Earth. A point sounds precise. Challenger Deep is not. Imagine searching for the lowest patch of ground in a valley, tens of kilometers long, except the entire landscape is hidden beneath nearly 11 kilometers of moving seawater. A ship at the surface cannot see which patch is lowest. It has to reconstruct the terrain using sound. Multi-beam sonar sends a fan of acoustic pulses downward and listens for their echoes. Directly beneath the vessel, the path is relatively straightforward. Farther toward the edges of the sonar swath, the beams travel at increasingly oblique angles through an ocean whose temperature, salinity, and pressure affect the speed of sound. Small errors in those calculations grow across 11 kilometers of water. The ship itself is rolling and pitching. Its exact position has to be known. Sea level is moving. Even local variations in gravity matter when researchers try to turn pressure readings into depth measurements accurate to within only a few meters. This is why modern surveys can disagree slightly, without any of them being careless. One expedition may cross the lowest terrain at a different angle. Another may collect denser sonar coverage. A submersible can carry a pressure sensor directly to the bottom, but then researchers must calculate depth from that pressure while correcting for the water above it and the local gravitational field. The deeper the measurement becomes, the more the final few meters depend on details that would be irrelevant almost anywhere else. During the 5 Deeps expedition in 2019, the research vessel Pressure Drop mapped enormous areas of the Mariana Trench with a full ocean depth multibeam system. The expedition also had something especially useful: instruments carried all the way down by the submersible limiting factor, and its landers could provide full-depth measurements of temperature, salinity and pressure, improving the sound speed profiles used to correct the sonar data. The resulting surveys helped refine a picture that had been developing for decades. The western pool of Challenger Deep is where many famous human dives have taken place, including the 1960 Trieste descent and later limiting factor dives. The central and eastern pools contain their own extraordinarily deep terrain. Rather than one smooth basin with a single obvious center, the bottom rises and falls by tens and hundreds of meters across distances that appear tiny on a map but become significant when traveled by a submersible. And this landscape is still changing. The trench exists at an active plate boundary. The Pacific plate bends downward here, and that bending fractures the incoming seafloor. Faults cut across the plate, steep slopes become unstable, earthquakes shake sediment that has slowly accumulated over years or centuries. Material can break loose and move downslope in submarine landslides, eventually spreading across deeper areas as sediment flows. Some of these events happen on scales no camera is likely to witness directly. Their evidence is left behind in the layers of mud. A sediment core can therefore function like a history book. Distinct layers may record periods when material suddenly arrived from farther upslope, rather than settling slowly from the water above. In a trench, gravity is constantly trying to move loose material toward the axis. This is one reason the deepest floor can contain more organic matter and more active microbial communities than a simple model based only on depth might predict. The trench is not merely receiving particles vertically from the ocean surface. Its walls can funnel sediment sideways and downward. A disturbance kilometers away can eventually affect what accumulates at the bottom. Challenger Deep is therefore both a geographical low point and a collection basin, but it is not the only extraordinary depression in the Mariana Trench. Travel roughly 200 kilometers to the east, and another deep region appears, Sirena Deep. It was identified through detailed mapping by the Hawaii Mapping Research Group during surveys around the turn of the 21st century, and was originally known as He-Marg Deep. It was later renamed Sirena Deep after a legendary figure from Mariana culture. Modern measurements place it only slightly shallower than Challenger Deep, around 10.7 kilometers below sea level. For most places on Earth, a difference of a couple of hundred meters would be enormous. Here it is the difference between the deepest and one of the next deepest known places in the ocean. Sirena Deep also helped reveal how misleading the old mental picture of the trench had been. The Mariana Trench is more than 2,500 kilometers long, yet public attention tends to collapse all of that geography into Challenger Deep. There are other basins, canyons and slopes throughout the system, many of which have received only a fraction of the attention given to its record-breaking southern end. A place does not suddenly become ordinary because it is 8,000 meters deep, instead of 10,900. From the perspective of exploration, enormous areas of the trench remain extraordinarily difficult to reach. This becomes obvious if we follow a submersible rather than a sonar map. A bathymetric map can display Challenger Deep on a single screen. A submersible experiences it at walking scale. Its lights may illuminate perhaps tens of meters of seabed clearly at any one moment. Outside that cone of light, the landscape disappears. If the vehicle travels several kilometers during a dive, the track may look impressive when plotted afterward, but compare it with a basin stretching many kilometers in every direction, and it becomes a thin line. Compare that with the entire Mariana Trench, and it is almost nothing. Even knowing exactly where the submersible is can be challenging. GPS signals do not penetrate thousands of meters of seawater. The support ship at the surface knows its own satellite position, but the vehicle below has to navigate using combinations of inertial systems, acoustic transponders, velocity measurements and communication with the ship. Sound takes time to travel. At Challenger Deep, an acoustic signal sent from the bottom can take several seconds to reach the surface. The position of the vehicle can therefore carry uncertainties that would feel absurd on land, but are normal when navigating nearly 11 kilometers below a moving ship. This has an important consequence for how we interpret every story from the bottom. When James Cameron described a sparse plain, he was describing the terrain his vehicle crossed. When Trieste stirred up soft sediment, Walsh and Picard were describing one landing site. When a lander records hundreds of amphipods around bait, it tells us what happened within a small illuminated patch at that location on that day. None of those observations can simply be expanded to describe all of Challenger Deep, much less the entire Mariana Trench. The scale difference is too large. If an explorer landed in the Sahara, drove several kilometers, and reported seeing no trees, that observation would be useful. It would not prove that no tree existed anywhere in North Africa. Hadal exploration works under an even greater constraint because researchers cannot look toward the horizon. Their horizon is the edge of the lights. This is one reason new dives can still produce surprises in places humans have technically visited before. A vehicle can return to Challenger Deep and travel across terrain no camera has ever seen. A lander dropped a few kilometers away can encounter a different sediment type or animal community. Better sonar can expose a small scarp that older mapping smoothed away. A new core can contain a microbial community unlike one recovered from the neighboring basin. Exploration becomes less about planting a flag at the lowest coordinate and more about building thousands of observations into a coherent landscape. Even the maps themselves can deceive us visually. Bathymetric diagrams often exaggerate the vertical dimension so that trenches are easier to see. Without that exaggeration, the Mariana Trench would look much broader and less like the narrow, terrifying crack familiar from illustrations. It is extremely deep, but its walls do not plunge 11 kilometers straight down like the sides of a well. The descent occurs across enormous horizontal distances. A vehicle traveling down the inner slope encounters gradients, terraces, scarps, and sedimented surfaces, rather than a single vertical drop. Remove the water, and the scale would be closer to an immense mountain landscape turned downward into earth. And the landscape does not stop at the trench axis. Look west, from the deepest floor, and the sea floor eventually rises through the Mariana Forearc toward the volcanic arc. This is the region above the Pacific Plate as it descends into the mantle. And once researchers began mapping and sampling it in detail, the Mariana story expanded again. Faults there act as pathways for fluids. Seawater carried into the subduction system reacts with rock far beneath the seabed. Material from Earth's mantle can be altered and returned upward. Entire mountains of mud can form where those fluids escape. Farther west, submarine volcanoes release heat and chemicals into the ocean. At one site, carbon dioxide exists as a liquid on the seafloor. At another, molten sulfur has collected in a crater beneath the Pacific. Challenger Deep may hold the record for depth, but the geology surrounding it reveals something more complicated than a record. The trench is only the visible edge of a system that reaches downward into Earth and then expresses itself again across hundreds of kilometers of seafloor. The trench is only the visible edge of a system that reaches downward into Earth and then expresses itself again across hundreds of kilometers of seafloor. To follow it, imagine the Pacific Plate continuing beneath the Mariana region after disappearing below the trench. The rock does not simply fall into an empty space. It carries part of the ocean with it. Water exists inside cracks and pores in the descending crust, and other water has already become chemically bound within minerals created by long interaction between seawater and rock. As the plate sinks, pressure and temperature rise. Minerals that were stable nearer the surface begin changing. Water is released from them and starts moving upward into the overlying mantle and crust. From the surface, none of this can be seen, yet those fluids help create some of the strangest environments anywhere in the ocean. Between the trench and the Mariana volcanic islands lies the Forearc, the region above the descending Pacific Plate before we reach the main volcanic chain. Here, deep faults can create pathways connecting seawater, mantle-derived rock, and the seafloor. Water enters fractures and reacts with a mineral called olivine. one of the major components of Earth's upper mantle. The reaction transforms the rock into minerals dominated by serpentine. The process is called serpentinization, and although the name sounds obscure, its consequences are remarkable. Serpentinization changes the volume and physical properties of the rock. It consumes water. It produces heat. Most importantly for biology, it creates extremely reducing chemical conditions that can generate molecular hydrogen and, through associated reactions, methane. In other words, ordinary seawater interacting with mantle rock can produce molecules that microorganisms can use as sources of chemical energy. The fluids created by this process can become extremely alkaline, sometimes reaching pH values around 12. That is far beyond ordinary seawater, which sits near pH 8. The chemistry would be hostile to most familiar organisms, yet those fluids do not remain permanently hidden beneath the seabed. In the Mariana Forearc, they can rise through faults carrying fragments of altered mantle rock with them. Over time, this mixture of fluid and crushed serpentinite can build entire submarine mountains. They are known as serpentinite mud volcanoes. Calling them volcanoes is slightly misleading because they are not primarily erupting molten magma. Instead, cold or relatively low-temperature mud, rock fragments and chemically altered fluids are being forced upward from below. Some of these structures rise kilometers above the surrounding seafloor. Their names, South Chamorro Seamount, Conical Seamount, Yinazao, Fantangisnya, belong to a landscape almost completely invisible from the surface. At their summits and flanks, fluids that have traveled through the forearc seep into the ocean. For years, one of the central questions was how far life followed those fluids downward. Finding microorganisms near the seafloor was one thing. Demonstrating that biological communities existed deep inside the serpentinite system was much harder. Drilling during scientific ocean expeditions eventually recovered material from beneath these mud volcanoes, allowing researchers to examine the chemistry and search for biological traces. And recent work has made the story considerably more interesting. A 2025 study of serpentinite mud from the Mariana Forearc found lipid biomarkers and geochemical evidence consistent with a subsurface chemosynthetic biosphere. The organisms themselves were not simply sitting in abundance waiting to be counted. Instead, researchers found molecular traces left by communities shaped by a difficult environment in which useful substrates may arrive only intermittently. Some of the fluids are extremely alkaline and poor in nutrients. Yet hydrogen and methane produced through water-rock reactions provide potential energy sources, and microbial life appears capable of persisting across these steep chemical gradients. This is an important distinction. The rock does not need sunlight to become biologically useful. Chemistry produced inside Earth can create the energy gradients life exploits. We are beginning to see the Mariana system differently now. Challenger Deep is the place where the Pacific Plate bends downward. The forearc is where some of the consequences of that descent return toward the seabed as fluids. Continue farther west, and another consequence appears above us. The water released from the sinking plate helps change the melting behavior of the mantle. Magma forms and rises. Eventually, it builds a chain of volcanoes, the Mariana Volcanic Arc. Some rise high enough to become islands, others never reach the surface. They remain submarine mountains, erupting, collapsing and venting chemicals into an ocean that conceals almost everything they do. When scientists began systematically exploring these submarine volcanoes with remotely operated vehicles in the early 2000s, they encountered scenes that seemed to belong to a different planet. One of the most memorable appeared in 2004 at a small submarine volcano called NW Ifuku. The remotely operated vehicle Ropos had been exploring at around 1,600 meters when its cameras reached an area of small white hydrothermal chimneys. Fluid was escaping from the seafloor. Around the vents were dense communities of mussels and other animals, but something else was rising through cracks in the rock, droplets that looked almost like bubbles. They were not ordinary gas bubbles, they were liquid carbon dioxide. The site became known as Champagne. Carbon dioxide behaves differently depending on temperature and pressure. At the surface, we know it mainly as a gas, and when cooled sufficiently, it becomes dry ice. But under the pressure found at NWI Fuku, carbon dioxide can occupy a liquid state. The droplets emerging from the seabed were about 98% carbon dioxide. They were buoyant, so they rose through the surrounding seawater, but the enormous pressure prevented them from immediately expanding into the kind of gas bubbles we would see at the surface. The effect on camera was strangely gentle. Small, pale droplets drifted upward from a volcanic seafloor, while hydrothermal fluids nearby reached temperatures above 100 degrees Celsius. When the ROV disturbed parts of the bottom, the flow of carbon dioxide increased dramatically, suggesting that a reservoir of liquid CO2 existed beneath the surface, probably trapped below a layer containing solid CO2 hydrate. The site was not simply geologically unusual. Life had gathered around it. Large beds of mussels occupied the vent field, along with shrimp, crabs, limpets, and worms. Bacterial mats covered parts of the seafloor. The animals were not feeding on sunlight transported somehow into the deep ocean. They were connected to microorganisms using chemical compounds from the vent fluids. Yet, even here there were limits. The extraordinary concentration of carbon dioxide changed the local acidity of the water. Researchers noticed something curious around the mussel beds. Despite enormous numbers of living mussels, empty shells were surprisingly difficult to find. One explanation was that after an animal died, the highly acidic conditions created by the CO2-rich water caused its calcium carbonate shell to dissolve unusually quickly. NWA Fuku had become both an ecosystem and a natural experiment in what high concentrations of carbon dioxide can do to marine life. Then there was Daikoku. Not far away along the Mariana Arc, cameras exploring this submarine volcano encountered another substance behaving in a way few people would ever associate with the bottom of the sea. In a crater, hundreds of meters below the summit, lay a pool of molten sulfur. Sulfur normally melts at temperatures a little above 100 degrees Celsius, and hydrothermal heat beneath the seafloor can keep it liquid. When that molten sulfur reaches colder seawater, its surface cools and hardens, forming strange yellow and dark crusts. Beneath them, liquid sulfur can remain trapped. YOV observations showed sulfur flowing, pooling and solidifying around the volcanic terrain. The site was so unusual that comparisons were made with Io, the volcanically active moon of Jupiter, where sulfur compounds help create its famously colored surface. Yet here the material was not millions of kilometers away, it was sitting beneath the Pacific Ocean, part of the same geological system that created the Mariana Trench. Champagne and Daikoku reveal why focusing only on Challenger Deep misses so much of the story. The deepest point is spectacular because of its pressure and scale, but the surrounding Mariana system contains environments that are chemically more energetic and, in some ways, stranger. Along the forearc, seawater reacts with mantle rock and produces hydrogen-rich, alkaline fluids. Along the volcanic arc, magma drives hydrothermal systems capable of releasing carbon dioxide, sulfur compounds, methane, metals, and heat. Each environment creates different opportunities and different limits for life. The connection between geology and biology becomes impossible to ignore. A fault determines where water can travel. A mineral reaction determines which chemicals are produced. Those chemicals determine which microorganisms can obtain energy. The microorganisms, in turn, support animals that could never survive there by waiting for food to fall from the surface. Even the location of a mussel bed can ultimately be traced back through that chain, to fractures in volcanic rock, to magma rising through the crust, to water released from a tectonic plate descending far below. It is an ecosystem whose first link is not the sun above the Pacific, but chemistry generated inside the planet. And that introduces a completely different way to think about life in the Mariana region. For most of Earth's surface ecosystems, the story begins with sunlight. Plants or microscopic algae capture solar energy, animals eat them, other animals eat those animals, and eventually the energy passes through an entire food web. Down here, there are places where the beginning of the story is hidden inside rock. Water moves through fractures, minerals react. Hydrogen, methane, hydrogen sulfide and other reduced compounds emerge. Microorganisms exploit the chemical imbalance between those fluids and the surrounding environment. To understand what happens next, we have to follow the energy rather than the depth. Almost every landscape we know begins with the same transaction. Sunlight reaches a leaf, a blade of grass, a microscopic cell floating near the ocean surface. Through photosynthesis, that energy is captured and stored in organic matter. Everything else follows. A whale can spend its life hundreds of meters below the surface and still depend on sunlight because the krill it eats ultimately depend on phytoplankton. A shark hunting in darkness may never see the sun while still living inside a food web powered by it. Even at Challenger Deep, much of the organic material consumed by amphipods and microorganisms began thousands of meters above in the illuminated ocean. Marine snow is sunlight arriving late. But the Mariana system contains places where that familiar chain is no longer the only possibility. Here, energy can emerge from chemical reactions taking place beneath the seafloor. The process is known as chemosynthesis, and the easiest way to understand it is not to think of it as life somehow eating rock. Certain bacteria and archaea can obtain energy by transforming chemical compounds. Depending on the organism and environment, those compounds can include hydrogen sulfide, hydrogen, methane, and others. The microorganisms use the energy released by these reactions to build organic matter from inorganic carbon. Functionally, they occupy the position that plants and phytoplankton occupy in sunlight-powered ecosystems. They create new biological material at the beginning of a food web. Once that happens, animals have several ways to take advantage of it. Some simply graze on microbial mats. Others consume organisms that have eaten the microbes. But some of the most remarkable animals have gone much further. They carry the microorganisms inside or on their own bodies. At hydrothermal vents and cold seeps, certain clams, mussels, and tube worms live in intimate partnerships with chemosynthetic bacteria. The animal provides access to the chemicals the bacteria need. The bacteria manufacture organic compounds that help feed the animal. Two completely different forms of life become parts of one functioning system. This is why a patch of seafloor receiving the right fluids can look dramatically different from another patch only a short distance away. Without seepage, the bottom may contain scattered animals surviving on material arriving from above. Cross an active fault where methane or sulfide-rich fluid is reaching the seabed, and suddenly there can be dense colonies of specialized organisms. The difference is not sunlight, temperature, or depth. It is chemistry. For a long time, scientists knew that this happened around hydrothermal vents and shallower cold seeps. What remained uncertain was how important the same process could become inside the hadal trenches themselves. There were clues. The Mariana Forearc already contained chemosynthetic communities associated with serpentinite fluids. About 80 kilometers northeast of Challenger Deep, researchers exploring the inner trench slope discovered the Shinkai seep field at roughly 5,550 to 5,860 meters. Clams occurred in dense colonies around a system associated with serpentinized mantle rock and methane-rich fluids. South Chamorro Seamount farther north also supported communities of mussels, clams, and worms around alkaline seepage, and later exploration found another serpentinite-hosted community at Asut Tesoru Seamount. These were important discoveries, but they were not communities living on the floor of Challenger Deep itself. That distinction matters. The Mariana system contains many environments, and finding chemosynthesis in the Forearc does not allow us to say that the same kind of animal community exists 11 kilometers down. For years, evidence from the greatest depths remained tantalizing rather than decisive. During exploration of Sarina Deep, researchers observed filament-like material attached to rocks at approximately 10,677 meters and considered whether it might represent chemosynthetic microbial growth. Chemical and dietary studies elsewhere suggested that seep-derived carbon might contribute to hadal food webs, but the technical difficulty of investigating tiny patches of seafloor under nearly 1,000 atmospheres of pressure meant that the larger picture remained uncertain. Then another part of the Northwest Pacific produced a discovery that changed the scale of the question. In the summer of 2024, scientists aboard the research vessel Tansuo Yihao began a series of dives into the Kuril-Kamchatka Trench and the Western Aleutian Trench using the full ocean depth submersible Fendouzja. These trenches are far from the Mariana Trench, so what they found cannot simply be transferred onto our map. But geologically, they share something important. They are deep subduction trenches where tectonic plates bend, fracture, and descend. On one dive in the Kuril-Kamchatka Trench, Fendouzja reached a site 9,533 meters below the surface. The cameras did not reveal the sparse scattering of animals researchers might expect at such a depth. Across dark sediment stood dense groups of slender tube worms. They belonged to the ciboglinids, animals whose biology is closely connected to chemosynthetic microorganisms. The site was appropriately named the deepest. At 9,533 meters, it became the deepest confirmed chemosynthesis-based animal community known on Earth. And it was not alone. The team continued diving. Across 23 dives, researchers found chemosynthetic communities during 19 of them. Some were dominated by tube worms. Others contained large concentrations of bivalves. One site at approximately 9,120 meters, called Wintersweet Valley, contained dense fields of two kinds of frenulate tube worms, extending through an area explored for around 2 kilometers during a single dive. When the expedition's observations were combined, the communities occurred across a remarkable 2,500-kilometer stretch of the Kuril-Kamchatka and Western Aleutian Trenches, at depths ranging from around 5,800 meters to more than 9,500. This was not a tiny biological curiosity around one unusual crack. Chemosynthesis was supporting extensive animal communities across enormous distances in environments where such ecosystems had rarely been directly observed. The chemistry explained why. Fluids rich in methane and hydrogen sulfide were moving through faults in the trench sediments. Isotopic evidence indicated that much of the methane was being produced by microorganisms deeper within the deposited organic material. Other microorganisms could then consume methane without oxygen, often working together with sulfate-reducing bacteria. That process generates sulfide. Sulfide-oxidizing bacteria can use the resulting chemical energy, and animals partnered with those bacteria gain access to a food source that does not require fresh organic material to fall directly from the surface. There is a beautiful circularity to this. Some of the organic matter buried in trench sediments originally came from the sunlit ocean. Microorganisms deep within the sediment transform it into methane. Tectonic faults create pathways that allow methane-rich fluids to rise. Other microorganisms consume those chemicals, and animals build entire communities around them. The geology does not create energy from nothing. It rearranges the pathways through which energy and carbon move, producing concentrated oases in places that would otherwise appear extraordinarily poor in food. What made the 2024 discovery particularly important for our story was where the communities occurred. They were strongly associated with faults along the trench system. The same tectonic forces that create hadal depth also fracture the seafloor. Those fractures can become plumbing. A fault is therefore more than a geological scar. It can connect buried chemistry to animals living on the surface. On land, we tend to think of faults mainly in terms of earthquakes. At the bottom of a trench, the same structure can help determine where an ecosystem exists. This forces us to be careful when returning to the Mariana Trench. The discovery in the Kuril-Kamchatka and Aleutian Trenches does not prove that an enormous tube worm field is waiting somewhere in Challenger Deep. No such community has been documented there. It would be poor science to turn geological similarity into a confirmed biological discovery. But it changes what is reasonable to look for. Before these dives, the deepest chemosynthetic animal communities were known from substantially shallower environments, and researchers had only limited evidence for how widespread such systems might be in hadal trenches. Now we know that dense chemosynthesis-based communities can exist at least 9.5 kilometers below the surface and can extend across vast sections of active subduction trenches. The Mariana system contains faults. It contains methane cycling. Its forearc contains serpentinite-hosted seep ecosystems. Its subsurface fluids contain hydrogen and methane, generated through water-rock reactions. Microbial communities associated with those processes have already been detected. None of this tells us exactly what remains undiscovered. It tells us that the search space has become much more interesting. A patch of apparently featureless sediment may sit meters from a fracture carrying chemically reduced fluid. A community could occupy a narrow geological boundary, missed by every previous lander. Remember how little of the bottom our vehicles actually see. A submersible can pass within a kilometer of something and never know it exists. At Challenger Deep, a kilometer feels close on a map and enormous through a viewport. This is where the limits of current sampling become impossible to ignore. The Mariana Trench stretches for more than 2,500 kilometers. Its hadal floor, slopes, scarps and forearc contain countless faults and sedimentary environments. Yet the number of locations sampled biologically remains tiny. Many dives have concentrated around famous deep points because depth records, engineering tests and scientific priorities naturally draw expeditions there. A geological feature only a few hundred meters across can easily remain unseen. The 2024 Northwest Pacific expedition demonstrated this dramatically. Researchers discovered communities stretching across thousands of kilometers in trenches that had been studied before. They were not hidden because they were microscopic. Some contained dense fields of visible animals. They had simply never been encountered systematically. And that may be one of the most important lessons the deep ocean keeps teaching us. Unknown does not always mean something impossible, monstrous, or completely unlike anything science has seen. Sometimes it is an ecosystem large enough to photograph, built from biological processes we already understand, existing in a place where nobody had pointed the right camera. Chemosynthesis makes that possibility particularly powerful because it removes one of the assumptions that once seemed to constrain deep life. An ecosystem does not always need a generous supply of fresh food descending from the surface. If geology creates the right chemical gradients, microorganisms can become primary producers in permanent darkness. Tube worms can gather around them. Clams and mussels can form colonies. Predators and scavengers can arrive. A local food web can develop around substances traveling upward rather than downward. More than 9 kilometers beneath the Northwest Pacific, that is no longer a hypothesis. It has been observed. In the Mariana region, the exact extent of such ecosystems at the greatest depths remains an open question, and that uncertainty should be preserved rather than filled with imagination. But we already know enough to overturn the old picture of the deep ocean as a world slowly starving beneath the productive surface. Some of its ecosystems receive energy through another route, carried by methane, sulfide, and hydrogen through fractures in the seabed. The sun may be thousands of meters away and completely invisible, while the planet beneath them supplies the chemistry. By now, Challenger Deep can seem almost disconnected from ordinary human life. No sunlight, no familiar pressure, no agriculture, cities or industry, and ecosystems operating through chemistry most people never encounter. It would be easy to imagine that whatever happens at the surface remains at the surface. The mud at the bottom tells a very different story. For most of this journey, reaching Challenger Deep has required almost absurd effort. Trieste needed a steel sphere with walls inches thick and thousands of gallons of gasoline for buoyancy. Modern submersibles depend on titanium, syntactic foam, specialized batteries, and support ships filled with engineers. Robotic vehicles have imploded trying to work at comparable depths. Acoustic signals take seconds to travel between the bottom and the surface. Yet some things created by humans have reached the hadal world without pressure vessels, pilots, or navigation systems. They simply sank. In 2014, scientists lowered baited landers into the Mariana Trench and recovered amphipods from depths between roughly 7,800 and 10,250 meters. These were the same kinds of small, scavenging crustaceans we encountered earlier, animals that gather quickly when food arrives on the trench floor. The researchers were looking for something that could not be seen on a camera: chemical traces inside their bodies. When the animals were analyzed, every sample contained persistent organic pollutants. Among them were polychlorinated biphenyls, better known as PCBs, and polybrominated diphenyl ethers, or PBDEs. These were not chemicals produced by the trench. They were products of the industrial world above it. PCBs were once widely used in electrical equipment because they were chemically stable and difficult to burn. PBDEs became common as flame retardants in plastics, textiles, and other manufactured materials. Those properties made them useful. They also created the environmental problem. A molecule that does not break down easily can remain in ecosystems for a very long time. Production and use of many of these chemicals were restricted or banned decades ago. But removing a chemical from factories does not remove the molecules already circulating through the planet. They can travel through the atmosphere and ocean, attach to particles, enter organisms and accumulate in fatty tissues. Predators and scavengers can then receive contaminants from the animals they consume. The surprising part was not simply that traces had reached a deep trench. It was how much had accumulated in some of the animals. In the 2017 study that reported the results, PCB concentrations in Mariana Trench amphipods were extraordinarily high, with the highest values exceeding those measured in some heavily industrialized coastal environments used for comparison. That result needs to be interpreted carefully. It does not mean Challenger Deep is generally more polluted than an industrial river, or that the water at 11 kilometers contains more contamination than water beside a factory. The researchers measured pollutants accumulated in the tissues of particular animals, and bioaccumulation depends on diet, species, chemistry, and the way contaminants move through food webs. But the finding destroyed any comfortable idea that extreme remoteness provides automatic protection. These animals lived kilometers beneath an ocean surface far from major cities, and their bodies still contained molecules manufactured by people. How those molecules completed the journey is less dramatic than a submersible descent, but perhaps more revealing. A pollutant entering the ocean does not need to fall directly from a ship above Challenger Deep. Ocean circulation can transport dissolved and particle-bound chemicals over enormous distances. Some pollutants attach to organic material. Plankton can absorb them. Small animals eat the plankton. Larger organisms eat those animals. Eventually, something dies or produces waste, and part of that material begins sinking. Marine snow, which we previously encountered as the food supply of the deep ocean, can also become a transport system for contamination. The same biological machinery that carries carbon downward can carry human-made chemicals with it. A dead fish falling through the water is not merely food, it is a package containing whatever that animal accumulated while alive. Once it reaches the trench floor, scavenging amphipods consume it, and substances resistant to breakdown can remain inside their tissues. The shape of the trench may intensify the process. We have already seen how steep slopes and sediment movement can funnel organic material toward the trench axis. Contaminants associated with that material can follow the same route. Trenches can therefore behave partly as sinks, collecting substances transported from a much larger area. That does not mean every contaminant entering the Pacific inevitably ends up in Challenger Deep, nor does it mean all hadal trenches accumulate pollution in the same way. The processes are complicated, and sampling is still limited, but the general pathway is no longer hypothetical. Contaminants have been measured in hadal animals. The isolation we imagined was geographical, not ecological. Plastic makes the connection visible. Unlike PCBs hidden inside tissue, a piece of plastic can sit directly in front of a camera. Japan's deep-sea exploration program has recorded debris across the ocean floor for decades, eventually building a database containing thousands of observations from research submersibles and remotely operated vehicles. Among them is plastic debris recorded in the Mariana Trench at approximately 10,900 meters. Think about the difference between that object and Trieste. Walsh and Picard spent years benefiting from engineering, planning and naval support to reach almost the same depth. The plastic required none of it. It was light enough to be carried, durable enough to persist and eventually dense enough, or weighted by organisms, sediment or attached material, to travel downward. By the time scientists arrived with cameras, some of our waste had already completed the journey. Plastic does not always remain recognizable. Sunlight, waves and physical abrasion can break larger pieces into smaller fragments before they descend. Organisms grow across surfaces and change their density. Particles become incorporated into marine snow or fecal pellets. Once small enough, plastic can enter food webs directly. Researchers examining amphipods from 6 of the deepest marine trenches later found synthetic fibers and fragments inside animals from every trench they studied, including the Mariana. In the Mariana samples, ingestion was recorded in animals collected at depths approaching Challenger Deep. Some of the recovered material resembled familiar textile fibers such as nylon and other synthetic polymers. Again, the interpretation requires restraint. Finding microplastic-like material in digestive systems does not tell us that every hadal animal is full of plastic, nor does it establish the precise health effect on each species. Deep-sea contamination studies face real challenges, including small sample sizes and the possibility of contamination during collection and laboratory analysis. But taken together with direct observations of larger debris and independent chemical studies, the pattern is difficult to dismiss. Manufactured material has reached full ocean depth. The routes downward are surprisingly varied. Some plastic begins denser than seawater and can sink. Other pieces initially float but acquire a biological coating known as biofouling. Bacteria, algae, and small animals settle on the surface, adding mass until the object loses buoyancy. Plastic can become tangled with heavier material or incorporated into aggregates of organic particles. Zooplankton can ingest tiny fragments and package them into fecal pellets that sink much faster than isolated particles. Larger animals can transport contaminants vertically by feeding in one part of the water column and releasing waste in another. Currents move material horizontally, while gravity and biology move it vertically. There is no single conveyor belt from a city to Challenger Deep. There are thousands of pathways, operating slowly enough that we rarely notice them. That makes the timescale important. The Mariana Trench does not need to be beside a pollution source to receive pollution. The ocean is connected. Water masses circulate. Animals migrate. Particles fall. Sediments move. Molecules manufactured on land can spend years traveling through these systems before arriving somewhere no human being has ever visited. Once they arrive, the cold and darkness that make the deep sea seem pristine can sometimes help preserve them. Many plastics degrade extremely slowly without strong ultraviolet radiation. Persistent organic pollutants are persistent precisely because ordinary environmental processes struggle to destroy them. A trench can therefore become the final destination for material produced during a human lifetime and potentially retain it much longer. There is a particularly uncomfortable symmetry here. The same processes that make the hadal ecosystem possible also expose it to us. Marine snow feeds the trench, but it can carry contaminants. Scavengers rapidly consume rare food falls, but that behavior can expose them to pollutants concentrated in the tissues of dead animals. Trench topography gathers organic matter, but it may also gather whatever is attached to that matter. Biological efficiency does not distinguish between a useful molecule and one manufactured accidentally into the food web. The ecosystem simply processes what arrives, and what arrives increasingly carries a signature of the surface. This does not mean Challenger Deep is a garbage dump. That description is dramatic but inaccurate. Cameras do not reveal a continuous carpet of bottles and bags across the deepest floor, and the limited number of observations makes it impossible to estimate debris across every part of the trench from a few encounters. Most footage still shows sediment, rocks, and animals rather than human rubbish. The stronger story does not require exaggeration. A single manufactured object at 10,900 meters is scientifically remarkable because of where it is. Persistent industrial chemicals inside endemic animals are remarkable because these species spend their lives in a habitat once assumed to be almost completely separated from ours. Microplastics inside hadal food webs matter because they show that the connection reaches beyond visible litter. The evidence is more unsettling when stated accurately. In 1960, humans reached Challenger Deep for the first time. By then, industrial chemicals were already spreading through the global environment. Decades later, when scientists finally learned how to sample the deepest animals carefully enough to examine their tissues, they found part of that industrial history preserved inside them. The trench had been participating in the human era even while almost no humans could reach it. This is perhaps the strangest reversal in the entire journey. At the beginning, Challenger Deep seemed impossibly remote. We descended through darkness, crossed the apparent limit of fish, entered microbial worlds hidden inside sediment, and followed tectonic processes into the mantle. Everything about the place suggested separation, yet the deeper we looked, the more connections appeared. Carbon from surface plankton reaches the bottom. Earthquakes move material down its slopes. Water enters the subduction system. Chemicals return through the forearc. Ocean currents transport matter across basins. And mixed into those natural cycles are substances that did not exist until humans manufactured them. We have influenced parts of the planet we have barely observed. There are sections of the Mariana Trench no camera has ever crossed, organisms whose genomes have never been sequenced, and microbial processes we may not yet know exist. Somewhere in that unexplored landscape, human-made molecules may already be present, which leaves an uncomfortable imbalance in our knowledge. We understand enough to know that Challenger Deep is connected to us, but nowhere near enough to understand everything happening there. We understand enough to know that Challenger Deep is connected to us, but nowhere near enough to understand everything happening there. And perhaps that is the most accurate place to end this descent, because almost every answer we have found has opened into another question. We know that animals live in the Mariana Trench, but we do not know every animal that lives there. We know that its sediments contain extraordinarily diverse microbial communities, but a genetic sequence recovered from a spoonful of mud is not the same as understanding how those organisms live, compete, reproduce, and exchange chemicals over decades. We know that snailfish have adapted to pressures that would destroy most familiar vertebrates, and we have a convincing biochemical explanation for why fish appear to disappear somewhere around 8 kilometers down. But the exact limits of complex life under pressure are still being tested. Even something as apparently straightforward as asking which species live at which depth becomes difficult when obtaining a specimen can require an ocean-going research vessel, days of preparation, and a machine capable of surviving pressures approaching 1,000 times those at the surface. Hadal biology is full of these incomplete pictures. Many species appear to be endemic, occurring in one trench or a small group of trenches and nowhere else we have sampled. That makes sense. Trenches are separated by enormous areas of shallower seafloor, creating deep habitats that can function almost like islands. Yet genetics has complicated that simple picture. Some microbial lineages appear related across geographically separated trenches. Hadal snailfishes spread between different Pacific trenches during their evolutionary history. Their gut microorganisms can share related lineages even when the fish populations themselves live thousands of kilometers apart. Somehow, isolation and connection are happening at the same time, and they do not operate equally for every organism. An amphipod, a bacterium and a snailfish, experience the geography of the Pacific in completely different ways. We still do not fully know how populations become established inside a trench, how often they exchange genes with populations elsewhere, or what happens when an isolated population disappears. Even basic life histories remain incomplete. For many hadal animals, scientists have only fragments of information about how long they live, where they reproduce, or how their young develop. A species may appear frequently at baited cameras, while most of its life happens somewhere the camera never sees. Bait itself changes the scene by attracting scavengers. A trawl selects for animals that can be caught by it. A sediment core samples organisms living inside a tiny cylinder of mud. Every method has a blind spot. This is one reason the unknown species of the Mariana Trench should not be imagined only as enormous creatures hiding beyond the lights. There may indeed be animals that have never been described, but the greatest undiscovered diversity is likely to include small invertebrates, microorganisms, and genetic lineages that would never appear in a dramatic photograph. A bacterium can be scientifically new without looking mysterious at all. Its importance may lie in an enzyme that works under enormous pressure, or in a metabolic pathway that changes how carbon or nitrogen moves through the sediment. We have already discovered microbial communities at Challenger Deep that differ from those in the abyssal water above them. We have found unexpectedly high microbial activity in hadal sediment, but identifying genes associated with a process does not automatically reveal how important that process is across the whole trench. To know that, scientists need measurements repeated across places, depths, seasons, and years. The Mariana Trench has almost none of that kind of long-term coverage. Geologically, the gaps may be even larger. Sonar has transformed the map, but a bathymetric map is the shape of the surface. It does not reveal everything moving through the rock beneath it. We know the Pacific plate bends and descends. We know faults fracture the incoming plate, and that water travels into the subduction system. We know fluids can return through the forearc and participate in serpentinization, while farther west water, released from the descending slab, contributes to processes that ultimately feed the Mariana volcanic arc. But tracing individual pathways through kilometers of crust and mantle is extraordinarily difficult. A fluid emerging from a serpentinite mud volcano may preserve chemical evidence of where it has been, yet reconstructing that journey is like trying to understand an underground river by analyzing the water at a single spring. The trench walls present another problem because they do not remain still. Earthquakes can destabilize enormous amounts of sediment. Landslides and turbidity flows can carry material downslope and rearrange parts of the trench floor. A layer of sediment that looks quiet today may record a violent event centuries ago. A future earthquake could change local habitats in hours, burying one community while delivering food to another. We do not have cameras watching most of the trench when these events occur. Often scientists arrive afterward and reconstruct what happened from disturbed sediment, bathymetry, and chemical signatures. This matters because the Mariana Trench is not merely a place produced by tectonics millions of years ago. The process is continuing. The landscape we are trying to understand is moving while we study it. Chemistry adds another set of unanswered questions. We know organic carbon reaches the trench from above, and that topography can help concentrate it. We know microorganisms break that material down. We know methane and hydrogen can be associated with geological and microbial processes in the wider Mariana system. We know chemosynthesis can support communities where reduced chemicals reach the seabed. But determining how much energy enters hadal food webs through each pathway remains difficult. The spectacular discovery of extensive chemosynthetic communities in other Northwest Pacific trenches has made the question even more interesting. Similar tectonic settings can create fault-controlled pathways for methane and sulfide-rich fluids. Does the Mariana contain comparable systems at depths or locations we have not yet sampled? Perhaps. But perhaps is where the evidence currently stops. The scientifically interesting part is not pretending that something must be there. It is realizing that environments we once considered incapable of supporting such communities have now been shown to support them elsewhere. That changes where researchers look and what instruments they send. Carbon itself is moving through several overlapping worlds. Some begins as carbon dioxide captured by phytoplankton at the surface. Some falls as marine snow. Some reaches the bottom inside carcasses. Some is buried in sediment. Microorganisms consume part of it and return carbon to dissolved forms. Other carbon can become methane deeper within sediments. Geological fluids introduce additional compounds from water-rock reactions. Trenches may enhance burial in some places while intensifying microbial recycling in others. To calculate the trench's role in the global carbon cycle, scientists need more than evidence that these processes happen. They need to know how fast they happen, over how much area, how they change after earthquakes, and how representative the few sampled sites actually are. That last problem follows us everywhere. Representative of what? Challenger Deep alone contains multiple basins. Serena Deep is different again. The trench extends for more than 2,500 kilometers. Its slopes cross enormous depth ranges. Its sediments differ with topography and material supply. The forearc contains serpentinite mud volcanoes. The volcanic arc contains hydrothermal systems. Calling all of this the Mariana Trench is convenient, but it can hide how many environments have been placed beneath one name. A sample collected from one patch at 10,900 meters cannot stand in for all of them. Neither can a successful dive. This is the fundamental difference between mapping somewhere and actually knowing it. Sonar can reveal a ridge without telling us what lives on it. A camera can show the surface without revealing the microbes centimeters below. A sediment core can reveal those microbes without showing what happens a kilometer away. A genetic sequence can identify a metabolic possibility without measuring how quickly that metabolism operates in nature. Every new instrument removes one layer of ignorance and exposes another beneath it. And obtaining those fragments remains difficult for reasons that have hardly changed since Walsh and Picard descended in 1960. Pressure still destroys equipment. Electronics still need protection. Navigation still becomes harder once satellite signals disappear at the surface. Communication is still limited by the speed and bandwidth of sound through water. Ships still cost enormous amounts to operate, and a research team may travel thousands of kilometers only to lose days to weather or technical problems. A lander deployed incorrectly can disappear. A corer can return without the sediment scientists wanted. A robotic vehicle worth millions can implode. A human-occupied submersible may spend hours traveling vertically before scientific work even begins. There is also a shortage that cannot be solved simply by designing better machines: time. A vessel may have several weeks in the region. The trench has existed for millions of years. Researchers must decide where to lower each instrument, knowing that every choice leaves thousands of other places untouched. This makes the phrase, "only a small fraction explored," more meaningful than it first appears. Exploration is not a percentage that eventually reaches 100 and becomes complete. The resolution keeps changing. Mapping the outline of Challenger Deep answers one question. Mapping it at higher resolution reveals scarps and basins that create new questions. Visiting one of those scarps reveals sediment, animals, and perhaps fluid seepage. Sampling the sediment reveals microbial communities. Sequencing them reveals unknown genes. Suddenly, the unexplored frontier is no longer kilometers away. It is inside a cell, recovered from a place already marked on the map. There may never be a moment when scientists can say that the Mariana Trench has been completely explored, because understanding is not the same thing as visiting. This brings us back to the surface, where our journey began. Above the Western Pacific, nothing announces what lies below. There is sky, moving water, and the horizon. Somewhere beneath it, the Pacific plate bends into Earth. Organic particles are falling through kilometers of darkness. Amphipods search the bottom for food. Microorganisms process carbon inside the mud. Faults provide pathways through fractured rock. Fluids move through the forearc. Volcanoes release heat and chemicals farther west. And in Challenger Deep, almost 11 kilometers beneath the waves, the deepest known seafloor waits in complete darkness, whether anyone is there to observe it or not. For most of human history, we did not even know its depth. In 1875, a weighted line from HMS Challenger gave us the first great clue. In 1960, two men finally reached the bottom. More than half a century later, vehicles began returning often enough for the place to become something more than a geographical record. Each generation of technology seemed capable of answering the mystery left by the generation before it. Instead, better technology enlarged the Mariana Trench. The first question was how deep it went. Then we wanted to know whether anything could live there. Then how life survived the pressure. Then what happened inside the sediment. Then how the trench interacted with the plate disappearing beneath it. Then, whether chemical energy could support ecosystems disconnected from sunlight. Then how pollution from our own world had already arrived. The unknown did not retreat into one final corner. It divided into smaller, better questions. That is why the Mariana Trench remains unsettling to scientists in a way that has nothing to do with imaginary monsters. We have already performed the spectacular part. Humans have stood, in the only sense possible inside a pressure sphere, at the deepest known seafloor. The technological question that once seemed impossible was answered on January 23, 1960. But reaching somewhere is a moment. Understanding it requires comparison, repetition, and time. It requires returning after earthquakes, sampling different basins, watching populations across years, following carbon through sediments, tracing fluids through rock, and discovering organisms whose existence cannot be predicted from a camera image. Almost 11 kilometers of water makes every one of those ordinary scientific tasks extraordinarily difficult. And perhaps that is what Challenger Deep reveals most clearly. The frontier was never simply the distance between the surface and the bottom. It is the distance between seeing a place and knowing it. We have crossed the first distance. The second may be far greater.